And I'll be your chairman this morning. There is a few changes in the program that is written. Unfortunately, Professor Mamba is not here to open this session and welcome everybody. So that will give us some time to interrogate the speakers before tea time. Simple house rules: ensure that your cell phones are unmuted or are on silent vibration mode so that you do not disturb the speakers. Like yesterday, I hope these sessions will be as vibrant as they were yesterday. So without further ado, let me introduce the first speaker. The first speaker is Professor Lucas Neyman. He's a professor at the University of Johannesburg within the College of Science, Engineering and Technology and within the Institute for Nanotechnology and Water Sustainability. He holds a PhD in physics from NMMU and he has an NRF rating of C1. His expertise lies in the physical processes, which are thermal and optical, optoelectronics, nano and microelectronics, and electronic control system development. Prof. Snyman has authored numerous journal articles, books, and holds 25 patents. And is also the Rainwater Chair at this university. Prof. Snyman, please join the floor.
Okay, good morning, everyone. Yes, I've been introduced. My talk this morning is on innovation and incubation at Unisa. And then also I will talk about research and commercialization, some suggestions. Okay. So, the outline of my presentation today is, first I will give a little bit of personal background, talk about real-life challenges that we have in South Africa, R&D approaches that I have followed, then the current innovation at our college, and I will give you an overview of prototypes that are ready for incubation, And I will then talk about technology transfer and incubation, and end off with a few conclusions. Okay. As you have probably heard, I have been a fundamental researcher myself in the area of semiconductors and processing. I've published about 100 articles and conference proceedings, journals. I did a lot lately. I've did a lot of applied research, and I have at the moment about 15 local patents, 6 international patents, and then also numerous grants. Lately, I focused on producing prototypes, mainly for industry and for application in South Africa. And from that approach, I actually got numerous grants lately from Unisa self, from government, and also lately a very big grant from Rand Water. So, what are the challenges in South Africa? I think in overall we have lots of a lack of job opportunities, but especially in the areas of water. Fresh drinkable water is a problem. Agricultural water, enough of that is a problem. There are extremely high levels of pollution, and we have contaminants entering our water reservoirs. Also in energy, our supply is intermittent, mainly due to the population size. These local suppliers cannot supply enough power for us. Then there's a lot of pollution as well, in terms of coal, and we have a transfer to green energies. In agricultural, I'm not going to highlight, I think most of you are in agriculture, you will know the challenges there. And the appeal, one of the big appeals I make today is that you should analyze your environment yourself and identify and define your own challenges. I repeat that because that's one of the big statements I want to make today. Analyze your own environment, define your own challenges. I was, about 5 years back, I was actually the director of the School of Engineering. And we started with a new model at that time, that was about 2015 onwards, where we said that, okay, in basic educations you have mainly three levels of division and training. At the top level, or let's start at the bottom, you have the undergraduate, and then in the middle there you have the level of graduate training, okay? And then at the top you have postgraduate training. And we reasoned at that time, we reasoned at that time that we can focus on applied research at the middle level, the graduate level, okay? we can focus on applied research at that level. Because you only need to do innovation and prototyping and demonstration for industry. You basically need skills. You basically need skills. And that can ideally be done by diploma students or graduate students. But then also you need postgraduate. Obviously there are always advanced problems, and those advanced problems can be addressed by the postgraduate level, postgraduate students. But what we also said is that there should be an interaction between all these levels. And I try to indicate that with the green arrows there. Typically, if you are a postgraduate student, you can become involved with teaching of graduate students and even diploma students. And even you can get paid for that. You can serve as student assistants or tutors, okay? And then also from the lower level up, you know, even from the graduate level to the diploma level, there could be tutorships and pathways. And then, as you may know, the undergraduate program is a big spinner of money. Okay. Whereas postgraduate training and even graduate training suffers from enough funding. So why not use then some of the profits that you make at undergraduate level and feed that into the graduate level and the postgraduate level. And similarly, you could feed back things like tutorship and teaching back to the undergraduate levels. And we've tried to implement this model. Now, if you focus on innovation and prototyping at the graduate level, as I said, where you basically need skills training, you need ideas and you need skills training, then that level is ideally a very good level to partner with industry, to set up partnership, to demonstrate your prototypes, your research results to industry. You can get involved in community projects, as I indicated at above there, or if you can't find a job in industry, if you graduate, you can even generate your own small and micro enterprise. And we actually started with this model. We had winter schools at the graduate diploma level, and we've built prototypes. That was about 5 years back. It was such a big success those times that we continued expanding the model even to today. And I'll show you some results, okay? And in this model, we can actually serve industry in one of the best possible ways. And we can create jobs in the country. Okay. So I want to move on to show you that all of this is actually the origin of all of this, or the postgraduate, plays a very important part. Right, I am from the Institute for Nanotechnology and Water Sustainability, and our focus is on water. We have high-tech, low-tech combination. We have rural community outreaches. And we are working in a number of areas. We have nearly 100 postgraduate students. It's one of the biggest institutes at the university. And we work mainly with on the area of sensors, water purification, conversion of energies, environmental applications, analytical chemistry, membrane technology, things like that. So, one of the things that we bought about 5 years back was a so-called high-resolution optical microscope. And this thing is standing there at electrical engineering. It has a resolution of sub-micron in the optical range. And we can take pictures at very high definition, all electronically displayed. And the big advantage of this is that you can do image processing. There are color information, and you can also do various analytical analysis with this machine. Some of the things that we did was to analyze bacteria. And there on the left-hand side, you can see a clear example. You have the dark areas, gray areas there. Okay? And that represents bacteria growth. But in between, we have added things like silver nanoparticles, and even some cases, some other stuff, gold particles. And some of this has antimicrobial action, and that there you can see that it causes white spots there, and that kills bacteria. So you can see the type of work that we are doing. Another area of research I was involved with is to start with a carbon electrode, and this is all done in solution. We add silver nanoparticles, those are the gray dots there on this electrode. And then you add to the solution antibodies. This is these orange structures here, which then bond with the nanoparticles because they have a very big surface area. And then we add an immunosensor, which is the gray blobs there. And all this together increase the affinity for PSA. Okay? For PSA. And we started experimenting with this procedure, and we did cyclic volumetric analysis. In other words, we look at the current, and we look at how does the current vary if you apply voltage to these electrodes. And from that you could analyze then, you can even 100% ascertain if the absorption actually did take place. And here we have two peaks here on the reduction side. You can see a peak there, reddish on the red curve, and then on the black curve we have another peak. And those peaks confirm certain processes, particularly this bottom one confirms that the PSA did in fact absorb. Okay, another interesting thing that intrigued me was that in the whole process, if you look at the optical blueprints, if you look at the optical prints in the various stages of the process, you can see definitely the green, the red curves, and the blue curves. As soon as the process occurred, there are a shift in these optical peaks. There you can clearly see these peaks are much narrower, they are much lower. So there's a shift here towards the ultraviolet. Also further on in the process, you get shifts in optical peaks. And these peaks are quite major, okay? And with time, the process carry on for about 10, 20 minutes, and eventually it saturates. And then we also did interference studies or selectivity studies. The voltage current measurements can uniquely identify the processes. So it just shows you that everything starts with fundamental research. Okay, we have been doing for a long time research in generating optical sources in silicon on chip. And here you can see that this is work that we've published. You can see that we actually managed to make a 100-fold, even 1,000-fold increase in the optical emission intensity. So everything starts with fundamental research. And then we looked at all kinds of possible applications. As I said, you can make a probe using your semiconductor technology. You have a source, and you have a waveguide, and you have evanescent coupling, and you can make a slot there, and you have your biomaterial, and then you have a detector. And this device, you can actually make at nanometer dimensions, very, very small. You can actually put it inside a needle. Okay. And the same process I showed you, we can also apply to detect breast cancer. Okay. We can detect air quality. You can detect bacteria. So this type of research could easily be expanded into many, many other microprobe sensors. Okay? And this is a typical idea that we have at the moment, is to make this all on chip, and then to have emission of your optical rays. This intercepts with the receptor layer. I call that chemical process I talked about as a receptor process. And then we could look in the reflection mode, or we could also look in the dispersion mode at the optical printouts. So it just shows you that everything starts actually with fundamental research. Okay. Now, if we go to skills training, we move down to the lower levels, as I indicated, to the diploma and the graduate level. You need skills training. People that knows electronics and processing, and they can build, and they can manufacture, and they can make prototypes. So one of the things that we did is actually to couple a laptop to a little microprocessor with various actuators there. I don't know if you can see, but there's a fan and there's a heater, and there are about 7, 8 sensors there. And all this you can install on a little board, and everything is controlled by a microprocessor, and on the laptop you get your plot out of all your graphs. So it took us some time to learn this technology, to establish it. And then we started to build prototypes. Okay? So at the college, I think in the last 5, 6 years, there are many examples of prototyping at the middle level, at the graduate level, at the diploma level. It just, and mostly this emanates from your research, your postgraduate research. And one prototype that one of the first ones that we developed was at Case or Ideas. They manufactured a reservoir here, and they added organic waste there and some water. And they left it in the veld, and this is a kind of bioreactor. It produced biogas. We could light up a stove there, and we could cook with it. And I think there has been many of these bioreactors been built already and distributed in the community. So this just shows you how fundamental research can penetrate to prototyping and to the community. Just one good example. How's my time doing? Yes, so we started also, I know that in the School of Engineering at Case that time, they were working on taking plastics, waste material, carbon, feed it into reactors, and then they add catalytic reactions to it. And they could make things like carbon monoxides, hydrogen gas. Okay? This is a form of energy, low-cost energy. And several of these things, I think, have already been distributed to communities. So it just, again, shows you how from fundamental research we can build prototyping. Okay, myself, we started with an idea to automate solar panels. Here you can see the solar panels. We've put a couple of motors to it, and then you could build long arrays of these, and the panel tilts with the angle of the sun. And we got a 20-25% increase in collection efficiency. Again, an example of prototyping and research that can be applied in the community. Okay, another example is we just took a piece of roof and we added black PVC piping to it in a certain array here, and later on we developed much more complex structures at a certain angle. The pipes are sitting at certain angles towards the sun, and just by that we could capture up to 10 kilowatt hour of thermal energy per day. If you drive at the back of CSET, you can actually still see one of these setups there. Right? 10 kilowatt hour in one day. If you multiply that with R3, it's about 30 Rand per day. Multiply it by 30 days a month, it's up to 400 or 500 Rand per month that a person can save in the community. Or even you can put it in your urban houses. You can capture energy, kilowatts of energy, and thermal energy. So this is much cheaper than solar cells. Solar cells, you pay about over 4 Rand per kilowatt hour. You know what is the cost of this? 30 cents per kilowatt hour. So it just shows you, if you use your mind and you do a little bit of management and prototyping, you can make major impacts. Imagine if we could mass produce this in South Africa. We could take away load shedding forever. Okay? So we're busy with that. We have one startup company in Pretoria. There you can actually see the guy there. And we've built this absorber structures. Now, another nice thing about this is that you can actually store the heat energy. You could store the heat energy in conventional geysers. And then you can supply all the hot water in your house for washing, for cooking, showering, everything. 10 kilowatt hour a day. What is even more viable is that we started doing research and ideas with using a new kind of solid state cell. We call it a thermoelectric cell. Where you actually have a big aluminium plate here, you put some of these cells in between, and then you have a cooling on the other side. Now, if you couple this thing to your hot water, you can generate electricity. There you can see a string of LEDs burning. So imagine at a house that you can capture thermal energy and you can also make electricity. So now you cut down your Eskom bills, right? So all these things are ideas that we had and that we implemented with prototyping at the diploma and the graduate level. We had a winter school. One of the projects at the winter school is build a cheap wind turbine with a high capture efficiency, okay? And we've built several of these wind turbines, and then we built more advanced. These turbines we erected in Soweto, or actually Lenasia, I think is another urban area there nearby. So we implement this prototype wind turbines. This student is getting his doctorate now. He continued with this. We've published 2 papers on it. And we are still trying to commercialize it further. But there are many other examples. And my time is running out. I'm not going into detail. But you can take your grid electricity and you can build energy managers that can optimally manage your energy inside your house. Or you can use a cell phone, and you can program your cell phone to switch on lights on and off only when you need it. Okay? Things like that. This was a small little micro tracker. Some of our later projects in the water area is to circulate water in a closed circuit system here, then use UV lighting and using titanium oxide nanoparticles to purify this water. There you can see the setup is working. And we used a microscope to actually test the water quality, and the results were actually very good. It was all done with students at the diploma and the graduate level. Okay, another one is one of the other problems is that the water levels is lowering. South Africa is getting drier. So we are more reliant on groundwater. But groundwater is normally polluted by salt and minerals. So we implemented membrane technology together with our microprocessor technology. We built these circulating systems. We don't have 100% success yet, but we're moving very fast. Okay. Okay. So, here is a reactor that we built for rainwater. One of the problems of rainwater is that the drinking water, to clean the water, they use a coagulant, a sticking together agent, then to clean up the water. But what do you do with the sludge? Right? The mud. What do you do with that? Near the fall dam, this is piling up. And it's actually poisonous. And they can't get rid of it. So they told Unisa to come up with research and to try and come up with a solution. So we built a reactor here to try and separate the sludge into different layers. And then we could use chemical processing to separate the sludge even further back to its original content. So that's another example of prototyping. Okay, what are the routes to technology transfer? I think, first of all, we should have more focus on applied research, even at higher levels, because the country needs applied research. Then you need protection of your IP, because all this is done at the university. So patenting, copywriting is important. Publication of your results is very important. It's part of marketing. Publication of your results. Advertising of the prototypes, prototyping, advertising it, inviting industry, talking to industry. Then to set up business plans, to come up with ideas, to look for partners. Okay? Then to license patents. We have a central sections at Unisa that only do licensing of patents, and they can license. Then you could seek some capital. If your idea is good and your prototype is good, you will surely get capital. You can either go and get a good job, or you could start your own company and you can make your own money. You create your own jobs. So all these are ideas. Now, just quickly to end off, these are some of the publications that we published. We published internationally in very, very reputed journals. Okay? And even today we get applause for some of these publications. IEEE Quantum Electronics in the USA. We started to demonstrate, you know what? We got millions of rand because our research was applied, it was prototypes, and it involved also postgraduate and undergraduate. We focused on group work. So we got grants. We got grants from the NRF. Okay? Then Unisa ISP funding, international source funding. DST government awards, up to millions of rand. We got a Rand Water Chair grant, 5 years, 5 million rand. Then the DVC announced niche areas in renewable energies. We got 3 million South African rand up front. Okay, I'm more or less finished. So we have one startup, and we are actually getting invitations internationally for collaboration. I've made a list there. China, Germany, France. Okay. We get invited to international conferences to serve on panels. Okay. And the conclusion I want to quickly make then to end off, it starts with identifying your own challenges in your own environment. Develop your own expertise. To do fundamental research, but also mostly applied research. To promote group work, undergraduate, postgraduate together. Interface with industry, talk to them. Market, talk to them. Write proposals. If they cannot give you a job, then you you go the entrepreneurship route, you create your own company. But we succeeded in getting contract research. Eventually, I think we can change. And I want to stress, I'm a born South African. We can change the world for a better place. Thank you.
Thank you, Professor Snyman, for interesting talk. Analyze your environment, you'll never be unemployed. That's the take-home message. Do you have any questions and comments? Any addition to the speaker's repertoire? Thank you, Professor Snyman.
Okay, thank you.
All right. The second speaker is Professor McIntyre, who is an associate professor in the Department of Life and Consumer Sciences, where he teaches courses in animals physiology, behavior, and ecology. He is an NRF-rated scholar with C2, and his research interests in and experiences in studying the ecology of vertebrates, particularly marine mammals in the Southern Ocean and Antarctica. His current research includes studies on the ecology of protected seals in Antarctica and otters in Southern Africa. Professor McIntyre is a species coordinator within the IUCN Otter Specialist Group and is active in various other scholarly roles, in particular as a co-editor-in-chief of a multidisciplinary journal within the polar community named Polar Records. Professor McIntyre, can you join us please on stage?
Good morning, everyone. Hope you can hear me. Thanks for the opportunity to speak this morning. So I've titled this talk, Understanding the Ecology of a Protected Species, One Student Project at a Time. And the reason for this is, as I was introduced, much of my own research is based around the Southern Ocean, working on seals. But over the past few years, we've started a couple of projects focusing a little bit more on local species, particularly otters in South Africa. And these have been all effectively student-led projects. So yes, I'm interested in the research, and I've sort of encouraged a lot of this, but basically what I'll be presenting today is an accumulation of projects that have all been different postgrad student projects, ranging from honors level right through to PhD level. And I hope the point of this would be to illustrate to you how individual, seemingly small projects can all contribute to a better and a whole understanding of threatened and protected species. So many years ago, I read somewhere in a book, I forget where it was exactly, that the progression of science can often be sort of compared to lighting a candle in a very dark room. So when you light a candle in a dark room, you sort of start making a little bit of light, and you create this sphere around you of unknowns. Which effectively generates a couple of questions. So what's outside of this that you can see? And then you start You start actually sort of investigating those question marks. So you light a couple of more candles, and you start seeing a bigger picture, right? But the bigger the picture, the more questions you're generating. So the larger the area is that you can sort of, that becomes unknown to you, and so on and so forth. So it sort of continues. And the point of it is sort of within science, one of the key and sort of, to me, one of the coolest aspects of it all is that the more work you do, the more questions you generate, and the more interesting things there are to explore. And basically, that's one of the take-home messages of this talk, is keep asking questions and keep generating new questions with your research. So just a quick outline of how I'm going to proceed. I'm first going to just give you a little bit of background to otters specifically. What you can see on the image over here is an African clawless otter. They used to be called Cape clawless otters as the common name, but over recent years we've finally wrestled that term away, as African clawless otters is a far more representative name, as the species occurs right throughout most of sub-Saharan Africa. And then I'm going to go through individual student projects and basically give you an idea of how one student project led to another and generated another question. Then we got a new student to ask that question and start investigating that. And then lastly, I'll end off with a couple of pointers of where we're headed from here. Okay, so African clawless otters, as I mentioned, they're distributed quite widely throughout sub-Saharan Africa. Let me just see if I can get a pointer. I'll just use my mouse here for those online. So you can see that their distribution is right throughout sub-Saharan Africa, except for effectively the Congo Basin, where they're replaced by another species of otter called the Congo clawless otter. And the species is one of the things that sort of led to a lot of my interest in them is because they're so widespread, they must show a lot of behavioral plasticity in terms of how they exploit their environments. So yes, they're always located and associated with fresh water sources, but these environments in which these freshwater resources occur differ substantially. They differ in terms of the prey that they offer the otters. They differ in terms of the physical structure of the water bodies, in terms of water flow, the water quality, all of these aspects. And yet, these otters seem to be able to manage in many of these environments. Having said that, there are many, many, many questions that we have about the species, and they're really, really poorly understood. So much so that if you start having a look at publications just over the last sort of decade of all otters that occur in sort of southern and sub-Saharan Africa, there are really only a handful. I think this is something like 23 odd publications that have included some research on otters. And as you can see, they're concentrated right in South Africa, where we've been contributing to quite a lot of this. I do wish to point out that there are two other species of otters that do occur in sub-Saharan Africa. Also in the middle here, these are spotted-necked otters. They have quite a wide distribution as well, and then the Congo clawless otter, as I mentioned earlier. So these, one of the key things is many of these studies have been really associated with some of the very basic ecology aspects of otters. And a great deal of the historical data that we have is really limited to, how can I say it, basic ideas around what they eat, and that's pretty much it. And those studies have been based on signs, so researchers would go out, because these animals are shy, you hardly ever see them in the field even though they're there. You rely on signs that they are present. So you go and you find a latrine, a place where they communally defecate, and you find some fecal samples which you can analyze, and from that you can start determining basic diets, for example. Or you just look for signs in terms of different environments and see where are there more signs than others. So we've tried to, over the last few years, try and sort of elaborate, build on that a little bit and start to get a better understanding of their ecology. One of the nice things about otters is, or one of the potentials, should I rather say, is that they are potentially great indicator species for water bodies. As a higher order predator in these systems, they are likely to be very sensitive to changes. So as these environments are changing due to anthropomorphic impacts such as pollution, climate change, etc., these species are likely to show big differences in terms of their numbers and how they use these environments. So we're trying to build enough of an understanding of their ecology so that we can effectively start using them as indicator species. So, one of the first student projects was about almost 10 years ago now. One of my first students, Rowan Jordan, he decided, well, what we need firstly is a little bit of a better understanding of some of the baseline ecology details in terms of what they eat and how flexible their foraging strategies are. How much do their diets differ between areas, between individuals, and so on. So what he did was, initially he did his honors work using a very straightforward means of assessing hard part remains of prey items in fecal samples. Here is on this photo just pointing to a latrine. So as I mentioned, otters have this behavior whereby they communally generate these latrines where they defecate en masse, and these are quite easy to find in the environment if you know where to start looking for them. And what he did initially was starting to look at what these prey remains were in these fecal remains. However, that has many limitations. So what he decided to do for his master's was to try and elaborate on this and start looking at other measures of diet, specifically trophic diet. And one of the cool ways one can do this is if you're able to get a couple of samples from animals, you can use stable isotope ratios from those samples in order to get an idea of what sort of trophic level the animal has been foraging on. So normally these are used, often people would be using blood samples, and blood samples are great, but the turnover of isotopes are quite fast in blood samples. So one of the really cool types of tissue that you can use, particularly in mammals, are whiskers. So otters have these long whiskers, and what these whiskers show, effectively if you subsection them into little sections, is a time series of what the animal was likely to have been eating over the time period that whisker grew. So what he did was he sampled a whole bunch of animals. He went about catching animals in different parts of the country and started analyzing these whisker samples for isotope ratios. Now, in particular, I'm not going to go into a lot of detail here just for the sake of time. I've got a number of other projects to run through. But on these graphs, what you see are isotope ratios of nitrogen-15 and carbon-13. Typically what we use is nitrogen-15. That gives you a trophic level idea of where the animal has been foraging. And if you have enough data for the underlying system, so in other words, if you've sampled potential prey items and you have an idea of what their stable isotope signatures look like, then you can start building a nice food web and try to figure out where these animals sort of fall out and what they're likely to have been feeding on. Anyway, but one of the key take-home messages that I want to show you here is if you look at these graphs, so the blue lines are nitrogen, which is that trophic level that I was talking about, and they run from the base of the whisker through to the tip. So the tip is the oldest part, so this is sort of running back in time, and the base is the nearest part, so that's the last part that was growing, illustrating what the animal was eating most recently. And what you can see is clear differences between individuals. Each of these plots represent a different individual. There are many more. I just picked out a few from the paper here just as illustration. And you can see for an animal like this very first one, you've got a near straight line where this animal is foraging basically on the same thing throughout. So it seems it's an indicator for us that that individual is at least specializing on a specific prey type. If you compare that to the next one, you can see this big variation where the animal was initially, a long time ago, foraging at quite a high trophic level, and then switching diets to something that was falling out at a much lower trophic level, and then returning to a higher trophic level more recently in time. So that, in turn, shows some flexibility. So you've got individuals that seem to specialize on prey items, and you've got individuals who seem to be quite flexible and maybe just sort of switching between what is available in that time that they're sampling. Moreover, there are differences in terms of the overall trophic level. So you've got other individuals like this one that's foraging at a lower trophic level than the one over here, but still specializing on a specific prey item. So this gives us some clues, and like I mentioned, if you know enough about the underlying system, you can start plotting out what the animal was likely actually foraging on. And so this is just an example from two animals that were sampled from a freshwater system around Dahlstrom, around a trout estate specifically. And what you can notice here is that at least one of the individuals here showed quite a large proportion of its diet was made up of the trout that is being stocked in these systems. So this in itself was perhaps not overly surprising, but surprising nonetheless. The reason why I say this is because clawless otters are thought, or were thought, to have been specialist crab feeding animals, basically. That they would be, even though they're present on fishing areas where fish occur, that they are typically too slow to catch, especially fast-moving fish such as trout. But this research, along with, we actually have some camera evidence as well of otters carrying trout around that they've evidently caught, showed that they are certainly consuming trout on these farms. Now we know also from just discussions with farm owners that they've suspected this, although they suspected mostly this to be a problem for spotted-necked otters, which co-occur with clawless otters on these properties. But we were unfortunately able to show them that actually both species are actually taking out their trout for them. So this led to the next question. So how much of a problem is this? So what you see on this image here, this is just a little signboard that I thought was quite appropriate next to a trout estate, one of the dams where people go fly fishing. And if you look very carefully, there's some remains lying over there of otter scats, of otter fecal material. So the otters sort of move around there, and the potential is there that they're really having a great time on these trout farms, that this is an easy food resource, and they're potentially exploiting this to quite a large extent. But we don't know how big the problem is. So this led to the next project that I'd like to quickly talk about, which was by another student of mine, Marie de Vos, who wanted to effectively get a handle on how big a problem is this. We know that the trout numbers in many of these stocked areas do seem to decline at higher rates than would be attributed to what the anglers take out themselves. And we also know that there are other predators there. We know that there are cormorants, for example, and we know that there are fish eagles, for example, that also take out a couple of trout at the same time. So how much of it is likely to be associated to otters? So we designed a little experiment whereby she did a mark recapture study on the trout that were stocked, in which she basically microchipped each little trout that was being stocked in controlled dams. And then the idea was, hopefully, we would be able to find back those microchips in otter latrines, and then started getting an idea of how many of those individuals that were tagged and stocked in these dams are evidently being consumed by otters, the tags passing through their digestive tracts and ending up in their latrines. Unfortunately, that part of the experiment just simply didn't work. We didn't find a single microchip in any of the latrines that we know of. There are multiple reasons why this could be the case. We're not sure. Potentially the damage caused to the microchips from the ingestion process was too much and they simply weren't functioning afterwards. Or potentially otters were, I don't know, coming across the microchips and spitting them out. Who knows? But we didn't. This was something that still needs a bit refinement, and we'll hopefully get there in time. But what she was able to do was, through the mark recapture process, from marking individual trout and monitoring how their numbers change, she was able to start modeling how quickly these populations decline over time. And that showed an interesting trend where the survival probability of fish was extremely low. Initially, over a very short period of time, the first week or two past stocking, survival probabilities were really high, but hardly any fish survived beyond a couple of months. Basically, the survival probability of any individual fish surviving in any of the two systems we tested to be over a year was 2%. So they weren't surviving. And this is in stocking systems where animals are placed back after capture by anglers. So they're not being removed by any other means. So we know that this is a problem. One of the next interesting things we wanted to know was how much of this, or how is this being managed by owners? What do they do about this? So she did a little study whereby she contacted a whole bunch of farm managers and owners and basically asked them, well, what do you guys do? Is this a problem? Do you consider it a problem? And how do you deal with it? And what was interesting or sort of eye-opening to us was that roughly half of the respondents reported that yes, they do try and manage predator populations on their properties, and of those, the majority, the means of sort of managing this was fatal shooting of individuals. So they would go and actively hunt out otters in the system. Now, otters are, at least at the time, they were protected species. They're listed on the threatened and protected species list of South Africa, even though they're officially not threatened by extinction yet. And so this was illegal, but they were quite happy to share with us that yes, it's illegal, but we don't seem to have another choice. So this is clearly a problem. These results together led to another study. So next question was, how do we deal with this? And one of the things we wanted to do was assess potential ways of mitigating this. And one of the ways is potentially just generating spaces for fish to hide from otters in the water system. Because these are open water dams which are artificially generated. Fish are stocked in there. They've got nowhere else to go. So an otter comes in and is able to either corner them and catch them, or they basically have just nowhere to run. So if you generate a hiding spot where an otter can't get into where the fish can enter, then the potential is there to start sort of making it a little bit more difficult, at least for otters to get to them. So an honor student of mine, Tracy Bush, did a little experiment whereby we tested basically one of the key problems around this was how eager fish would be to use structures, artificial structures that are entered into these dams. We've had discussions with a couple of international partners, and they said, well, one of the problems is fish are a bit stupid. They don't like to enter artificial structures. So if you put it in there, they're not likely to use it. So we tested this. And this seemed to work. So we've got a couple of structures that we're working on, and hopefully in time we'll start rolling this out in the field. For time, I'm going to go a little bit faster because I think I'm a bit behind. So one of the other things we wanted to know was on these artificial areas, so particularly these triad estates, we noticed that there are quite a lot of otters, and we noticed that they seem to be doing quite well. But we didn't know how many there are. Are there really so many more there than other areas, and how are they doing in terms of their overall health? So another student of mine, Tapiso, did a study whereby she assessed firstly, the numbers of otters present on these areas. So what she did was we used camera traps to effectively assess the population density of otters. This is a bit of a complicated process. It takes quite a bit of modeling, but she did a really good job. Basically what we do is we set up an array of camera traps along water systems, anywhere between 20 and 30 camera traps at a time. We leave them out for a couple of weeks, and we get an idea of how many animals there are that pass through that area. A second part of her study was assessing population level stress in these populations. So what she did here was she looked at fecal glucocorticoids, glucocorticoid metabolites rather actually, and this is basically a key indicator of how stressed animals are. So she did a little validation study to indicate what background levels and which glucocorticoids are useful to use, and then went and sampled a bunch of really fresh fecal material from different sites and compared those between one another to get an idea of basically how stressed these populations are. What she found, firstly, that yes, the population density at these triad estates was sky high. So if you compare, just in this little table over here, Millstream Farm is the triad estate, and these are two control areas. And you can see the density here is roughly 8.3 animals per square kilometer. Compare that to 2 and less than 1 animal per square kilometer in natural areas. So yes, there are lots of animals, but are they happy? That seems to be less clear. So what we've shown here is, again, in these transformed areas, these triad estates, these fecal glucocorticoid metabolites, so basically the levels of stress hormones that are present in this population, is substantially higher than the natural areas. So we don't know what the reason is behind this yet. This is another thing that we will need to try and investigate in time. It could be that because there are so many animals around, that the density is so high that they're basically stressing one another out. That from a social structure perspective, they're not adapted to this. This is not a healthy system for them. Even though there's a lot of food and it's attracting more of them, it's creating a lot of stress in the population. What the downstream effects of that are is also still unknown. Okay, but what are the natural population densities? So we've had those two controls, but we don't know what the natural variation is between other populations. So what we've done is we've continued doing more camera trapping surveys around different sites across the country. Candice Lewis was an honours student a couple of years ago who then started assessing, or at least modelling, a lot of the data that we've been collecting over the years. And what she did was modify some of the models we've been using and start incorporating more of them to try and get an idea of how population densities differ between various protected areas that we've actually been sampling. So what the natural sort of variability would be. So we've sampled, so what she did for this paper, which was recently accepted in a journal called Oryx, she compared areas where we sampled in the Northern Cape, Western Cape, as well as Eastern Mpumalanga and KZN. And these areas, what we'd found here was that there's quite a lot of variation in densities between these populations. So don't pay too much attention. There's a lot of detail on these tables. I just want to illustrate two columns here. This is two different models, a random encounter model and a distance sampling model over here. And what you can see, the densities here for these sites vary anywhere between less than 1 animal per square kilometer up to about 4 animals per square kilometer in a natural setting. So 4 animals is really quite a high density, but this seems to be the general sort of trend that we're seeing between areas. We've carried on and done even more surveys after this that was not included in her study, particularly including sites in the northern parts of Namibia, around central parts of the Free State, Golden Gate, and also lower parts of KZN. And while many of these we haven't quite processed yet to get the actual density models, a basic comparison of what we would refer to as a relative abundance index seems to illustrate again that there are some areas that are surprisingly very low in terms of numbers of otters that they support. So what you see here, this relative abundance index is, like I mentioned, it's simply a relative number. It gives you an idea of green is basically lots of otters in the area, blue is seemingly somewhere average, and red is very few. And what you notice here is some of these areas, notice that all of these are protected areas. So Golden Gate, for example, this is Mtamvuna Nature Reserve in southern KZN. That's Babwata National Park in Namibia on the Okavango River. All have very, very low densities of otters, surprisingly so. You'd think somewhere along the Okavango would be really high levels of, or high numbers of otters. We don't really know why as yet. We have a couple of hypotheses. One of them for some of the specific sites, specifically for Babwata, is that there are potentially too many crocodiles there for otters to really do particularly well. So a lot of this work, again, that work specifically fed into another student project where she not only monitored she looked for signs of otters not only along the Okavango but also along the Kaneni rivers in the northern parts of Namibia, and effectively showed that part of Namibia seems to generally support very few otters, far fewer than what we would expect, which is concerning. We don't really sure what the reasons are. For some of the systems, particularly along the Kaneni, it could be that there's a lot of human impacts. There's a lot of fishermen around there, a lot of people living off the river, which potentially impacts otters as well. But this is something that needs more work as we go. Okay, one of the things that we found particularly along these latrines, is some really interesting behavior. So when otters come along, they do these little jiggle dances when they we call it spraint, where they basically paste the area with an anal jelly, which is effectively something that we're not sure about the purpose of. So what we wanted to do was try and get an idea of what does it mean? What's the social implications of marking territory like this? So I had another student, Stephanie, who started investigating some of this. One of the key things we wanted to find out was, firstly, what are the sort of physical characteristics of the areas that otters choose, and what are the chemical signatures of these otter latrines, and what can these two measures tell us about the potential role of latrines and scent marking for otters. So what she did was she basically modeled the occurrence of these latrines, the physical characteristics of the areas in which they occur, and there was a clear selection for open areas where they were seemingly picking areas that were more conspicuous, potentially for other otters. So maybe advertising that they're in the area. There was also a selection for relatively wind-still areas. If you compare the wind speeds, average wind speeds for areas that had latrines and areas that didn't, areas without latrines had higher wind speeds. So we hypothesizing that potentially aids in scent retention. When she analyzed the compounds, she identified 34 compounds, volatile organic compounds, in these latrines, of which most of them we don't really know what the role is, so we can only go and compare them with what's been published in literature for other species. But what we were able to tell is that 9 of these compounds are known to play roles as sex pheromones or as reproductive status indicators in other species. So we're hypothesizing that potentially these latrines are not necessarily only playing a role in terms of territoriality, for example, or communication with other species, but rather as an important role in reproductive behavior potentially, as in animals are advertising their reproductive status. I'm a little bit over time, I'll be quick. Okay, so where to from here? From here, some of the things that we're really interested in exploring includes things like impacts of poor water management practices and infrastructure and how that impacts on otter populations. So this is just a clip of the Jagersfontein mine disaster from a couple of years ago. This happens to be just upstream of one of our field sites at Kalkfontein Dam in the Free State, where we've been monitoring otter populations. So there's some data coming forth from that, hopefully in time, which will tell us a little bit more about impacts associated with this mining disaster. Another issue that we're keen to explore, and I have a student that started recently looking at this, are urbanization effects. So recently, particularly in Cape Town, around the waterfront area, around Greenpoint, particularly since lockdown, since COVID lockdown times, otters have become a little bit of a pest. So they've become really habituated to humans. They are sometimes seen on pathways, interacting with humans, interacting with dogs, as you can see in this little video clip. And there have been instances now of people also being bitten by otters. So this is becoming a bit of a management issue for the city of Cape Town. We've been collaborating with Two Oceans Aquarium, who've been monitoring the situation a bit more. And we're hoping to start getting an idea of, well, how are these animals, why is this happening? How are they adapting to the environment? And what are the potential management implications associated with this change in behaviors, or this sort of urbanized behaviors. So they look cute here, but they're not such cute animals in terms of being that friendly necessarily. And then lastly, we need to do some more validation. Our camera trap-based density estimates are good, we think, but we don't really have the background information. So the next step from here is we want to do genetic work based on the fecal samples to get an idea, do basically a mark recapture of genetic samples to get a better, stronger idea of population densities to which we can compare our camera trap data. So 2 take-home messages for students: don't underestimate the value of your research. It contributes to more questions, and you need to embrace those questions. As you go, that gives you a much general, or as you start putting together what seemingly to you is your own small project, with the next student's small project, with the next student's small project, you can start generating a nice whole picture of what's going on. And with that, I'd just like to thank all the students who are involved and sponsor or people who have funded the work, and to you for your attention. Thank you.
Thank you very much, Professor. Any comments, questions?
So I'm Jasper Rees, I'm a sort of DNA person. In your last sentence you said genetic work, so we're going to have coffee together. But for the audience, can you give us a sense of how long the whiskers take to grow, and what you didn't put a time frame on your graphs? Do you know how long that reflects?
That's a very good question. Yeah, for those online, if you didn't hear, the question was related to the time frames in which whiskers grow. Unfortunately, we don't know. We have some ideas. It's likely a couple of months, just based on what we can see from captive animals, but we haven't actually gone and measured those whisker growth rates. And the interesting thing is whisker growth rates in other animals aren't linear. So they tend to, we've measured this in seals. This is sort of where it started with, we've done this with elephant seals before. And with elephant seals, that regrowth, if you basically pluck a whisker and you allow it to regrow, it grows much faster initially and then it slows down. So you need to account for this sort of, if you want to really start getting a timestamp. So for some animals, we know what that growth rate is, but for otters, we're guessing at the moment. Yeah.
Thank you very much. Thank you, Professor.
Sure. One more question? No?
That will be the last one. I feel like tea.
Thank you, Prof. For me, it's just a question out of curiosity. Prof, do you think that Congo claws are the hybrid from Cape claws, otters?
So they were, for a long time, they were recognized just as a subspecies. Whether it's a hybrid or not, that's unlikely. It seems the genetic work that has been done suggests that it's a separate species, and they seem to have this sort of reasonably clear delineation where really that sort of Congo basin, there are no African clawless otters. So there's no overlap in distribution as far as we're aware. But yeah, it's one of those Congo clawless otters are, if you think we know a little about African clawless otters, we know practically nothing about Congo clawless otters. So it's a good question.
Yeah, okay. And then, sorry to follow up on another one. Maybe I didn't hear the question from Prof. Can you determine the age using the whiskers?
The age of the animal?
Yes.
No. Because whiskers, so whiskers are replaced naturally. They grow and they do fall out as animals get older, and they normally, most likely, sort of a typical whisker would be no more than a couple of months old. So it can't tell you much about the age of the animal itself.
Okay, thank you.
Thank you, Professor. Yeah, thanks. The last speaker is one from the sponsors. Is Letitia Dix, sales specialist for the clinical and transitional research portfolio at Whitehead Scientific. Her title, What You Are Missing Matters, an introduction to sequencing with Oxford nanopore technology. Letitia, the floor is yours.
Okay, good morning, everyone. Thank you, and sorry for disrupting your tea time, but I promise this will be a very exciting talk about a very exciting technology from Oxford Nanopore Technologies. As soon as we get the slides going. No? Okay. Let us do this. It should be fine now.
You're supposed to duplicate, okay?
No, don't worry. I'm going to do it like that. Okay, wonderful. So thank you for the introduction. I am Letitia Dix. I am specialized in research in next generation sequencing during my master's degree. And for today, I'm actually going to give you a talk about a third generation sequencing technology and to show you basically what you're missing in your current next generation sequencing matters. So this is going to be a general overview of Oxford Nanopore technology. And today I also brought along one of the little sequencers that we have available from Oxford Nanopore technology, and it's called the MinION sequencer. This is a wonderful infield sequencer, and I'm going to talk a bit more about it throughout the process of today's talk. So the main idea of this talk today is to give you an idea of how you can generate ultra-rich data for answering questions with impact with genetic data. So how does Oxford Nanopore Technologies work, and how does it actually differentiate itself from all the other technologies, the sequencing technologies out there? So the main idea of Oxford Nanopore Technology was to democratize sequencing, which we as people on African continent know is very important. So this is for anyone to sequence anywhere, anything. That is the basic idea of a sequencing technology. So it's very accessible, it's affordable, and it's very easy to use. So what actually happens with this technology is you can generate real-time data to actually gain rapid insights into your research questions. It's very scalable. So as you can see, it can go from a very portable sequencer that can be used in field all the way up to the bigger instruments called the GridION instrument or the Promethean instrument. And all these instruments can actually be used at sequencing cores, generating up to 14 terabytes of data in a sequencing run. The other amazing thing about this technology is it is read length agnostic. In other words, it's not just a long read technology. It can go anywhere from 20 base pairs, sequencing 20 base pairs, all the way up to 4 megabase pairs, 4 million base pair reads. So it's also the gold standard for methylation sequencing data. And I'll go a bit more into detail on that and why the technology is able to do that in the next couple of slides. Another amazing thing is this technology can actually give you a chance to deliver all genetic variations. So in one experiment, you can access information on SNPs, structural variants, you can do assembly, phasing, and get full methylation data as well. So where has this been used already? So in the life sciences research, it's been used to basically increase our understanding in a lot of fields of research. especially in human genetics and cancer genetics, so oncology, human disease, infectious disease, reproductive health overall. Then for plants and animals, and more importantly, microbial organisms, this has been a great research technology. And I think for particular interest for everyone here is the amount of antimicrobial resistance research being done on this technology, as well as wastewater monitoring being done on this technology. So just to show how versatile this sequencing is, the MinION has actually been used to sequence in the International Space Station already, all the way through to the polar environments and everything in between. So there's been a lot of research on Ebola outbreak, SARS-CoV-2 also being done on this technology. And then at Whitehead Scientific, we've also been doing our own little experiments, moving from different universities and different research institutions to actually show how the technology works, and also to show how easy it is to implement in your own research facility. So at the heart of Oxford Nanopore is the nanopore. So basically, you're going to have a flow cell, and on this flow cell is where this picture comes in. This flow cell has a membrane, which is here depicted in the gray part. The membrane is then distributed with a lot of these nanopores that are embedded within the membrane. So the gray part is actually electrically charged, and what happens is when we have a DNA or RNA molecule moving through the nanopore, it actually disrupts the signal, the electrical signal that we have within the membrane. And what this does is this generates these little squiggles that you can see over here when my mouse is back. There we go. These little squiggle signals over here. And through AI technology, this actually gets read as your base pairs. So your A, T, C, Gs are being read in real time. And this is being done at 400 base pairs per second for a DNA strand. The other great thing about this is you can do DNA or RNA as a native DNA or RNA strand. So there's no need to do additional PCR steps, which will induce PCR biases, which therefore you won't capture your methylation data. So in other words, the squiggle signals can also show when a base pair is methylated so that you know what your methylation data is as well. So that basically gets included with your sequencing run. You also have the option of still doing amplicon-based sequencing approach. So the basics where you do your PCR targeted approach to find your regions of interest, and you can sequence those amplicons as well. Of course, then you're not going to get any methylation data because you did a PCR step, which is going to introduce those PCR biases that I spoke about. So it is an end-to-end platform. It includes technology for the library preparation part of your steps, the sequencing, which is the sequencing platform, and the flow cell that you're going to need. It doesn't need any other reagents to be included except for the library prep and your flow cell. And then it also has a bit of an analysis pipeline as well for our wet lab scientists to use. So just to end off that section, it's one sequencing platform that can allow you full biology. So SNPs, indels, methylation, structural variation data, phasing, and assembly. You can do anything from 20 base pairs all the way up to 4 million bases, depending on the DNA that you input into your sequencer, that is. It's scalable all the way up from 2.8 gigabytes of data that you can generate to 7 or 14 terabytes of data. And it's a very simple, accessible workflow with real-time data analysis, and the upgrades for the software is also included. So the real question that sometimes gets asked on this technology is, what is the accuracy? Well, here are some headline numbers on the accuracy of the sequencing instrument. So for simplex sequencing, which is basically just sequencing the one DNA strand, you're getting a 99.6% rate of accuracy, which is a Q24 score. For duplex, that would be if it sequenced the one DNA strand and automatically pulls in the other DNA strand, you're already getting Q31 and up accuracies. Now, the important thing to know here is this is raw read accuracy. As soon as we move on to the assembly accuracies, like with human and bacterial, we're getting around about Q40, Q50 accuracies within this sequencing technology. So library preparation options, which kits are available from Oxford Nanopore. Basically, what you're doing with Oxford Nanopore is the library preparation is there to add a motor protein. This motor protein is this little part that you see here in purple. This is actually a helicase. And what this helicase does, it actually binds to your nanopore so that it feeds through your DNA or double-stranded RNA or RNA. The other thing that this helicase does is it automatically also unwinds your double-stranded DNA so that it moves through the sequencing pore. Now, the reason the accuracy of Oxford Nanopore has increased so much over the years is they actually took the nanopore and changed the structure of it, in the sense that there are two regions now embedded in the nanopore that actually reads your data and or reads the base pairs going through. So instead of where previously there was only one region where it read your A, T, G, C regions, now there are two parts in the nanopore, highly increasing the accuracy of the technology as well. So we have options for DNA and RNA sequencing. Like I said, you can sequence the native DNA strand or native RNA strand. There are options for simplex sequencing, in other words, so sequencing one sample at a time, and you can also pool samples together. So this means you can pool around about 24 samples together in one sequencing run, drastically reducing the cost, or 96 samples can be multiplexed together onto one flow cell for a sequencing run. There's two options available on the DNA side, a ligation-based approach and the rapid barcoding approach, which is a transposase they use to add the adapters. The rapid kit is the one that has always been known as the fieldwork kit, reason being it takes about 10 minutes to generate a library and load it onto your sequencer. So what devices are available, the different sequences that we have available from Oxford Nanopore? There is a high range from what you need for your specific throughputs. So for the Flongle, we have a Flongle flow cell, which is basically a flow cell that can generate around about 2 gigabytes of data. This is perfect for your small virology type of research, smaller bacterial genomes that you want to look into, or just for general QC steps before you load onto a larger flow cell at the end of the day. Also, the MinION flow cell, this is an amazing flow cell that generates around about 15 to 30 gigabytes of data in a sequencing run. The amazing thing about this is the flow cells can actually be washed and reused. And I'll go in a bit more detail about that as well. So the MinION is this little guy that I have available today, and I'm also going to just quickly run through the other ones as well. As you can see, it's completely scalable. MinION can do one flow cell at a time. Your GridION can do 5 flow cells at a time or separately, which is ideal for student projects where there are different people needing to do different sequencing runs. This means that one person can start their sequencing run, and the other person, as soon as their library is ready to load, they can also start it on the same instrument as well. So this is just an idea of what the flow cells look like. The Flongle flow cells are one-time flow cells, that one-time use flow cells that need a little adapter to fit into our little sequencer at the end of the day. The MinION flow cells, as I said, anywhere between 15 to 35 gigabytes of data that you can realistically generate out of this. And here's just a picture, but of course I have a live thing here as well for you to see. So you're more than welcome to afterwards visit me at the stand to actually have a bit of a look on how this technology looks like. Like I mentioned, the GridION, 5 MinION flow cells that can go onto the system. And the nice thing is the compute is already included within the system. So no need to buy an additional computer. It will only need a screen and a mouse for you to operate the system. The Promethean flow cells, these flow cells are very powerful. They can generate around about 100 to 200 gigabytes of data, and they're basically as small as a Wilson toffee. So this is amazing for your larger throughput projects. If you're looking at plant genomes, human genomes, this would be the flow cells that you would probably be looking at. This is just to show the Promethean family, the scalability thereof. With the Promethean sequences, you have a Promethean P2, which can do 2 flow cells at a time, the 24 Promethean flow cells at a time, or 48 flow cells at a time. And for this reason, that is why you're getting around about 7 terabytes of data for the P24, 14 terabytes of data for the P48s. Another wonderful thing is you can actually use the GridION and add a P2 solo to it, and they will use the same computing power that is on the GridION device, which means you actually, as researcher, have accessibility to using the Flongle flow cell for lower throughput experiments, the MinION flow cell for medium throughput experiments, as well as the Promethean flow cells for your bigger research projects where plant genomes are needed or human genomes are needed. This is also just a nice slide to basically see where the size of the genomes fit in to be sequenced with what sequencing platforms as well. It's a very bespoke technology in the sense that it uses the same chemistry for all the platforms. The only difference is the flow cells have more nanopores embedded, and that gives you more data at the end of the day. Another few unique things to Oxford Nanopore technology is something they call adaptive sampling. Now, what adaptive sampling is it actually allows you to enrich or deplete for regions of interest. So it's basically a target-based approach that you can use for your sequencer. And how this works is you can actually include a bed file, and in that bed file you can state which sequences are of interest and which are not. What would happen then is your nanopore would start sequencing the regions of interest. If it sees it is a DNA strand that is not the region of interest, it will reverse the current and push out that specific strand to not sequence it anymore. And then in that way, you're actually capturing the data of interest, and only that strand gets sequenced for that specific research project. The flow cells, like I mentioned, can also be reused. And the nice thing is, because you're doing real-time sequencing, if you see something goes wrong with your sequencing run, you can instantly stop the run, pick up the library, load it into a new flow cell, do a couple of wash steps if you feel that is needed, and reload it into a flow cell and start the sequencing run again, offering you complete flexibility when you sequence. Then regarding the wash that I spoke about, you might be wondering, but surely there's going to be something left from a previous library run. Well, with a wash step, there's actually a kit that removes 99.9% of your template molecules from your previous library. And that you can see now nicely without washing on the right-hand side versus with washing, how the flow cell actually depleted those previous libraries so that you can load a new library run as well. Then, just to end off the data analysis part of things, Oxford Nanopore also provides you with data analysis pipeline as well. Firstly is the Minnow software, where you can control your sequencer to actually tell it that you're going to need methylation data, how long you want the sequencer to run, at what time you want it to stop when it's generated enough data. Then also, that's going to be for raw data acquisition, general QC, that software is going to be used. Then we also have something called the epitome pipelines. Epitome pipelines are included when you purchase one of these sequencers. And the great thing is, specifically for microbiology, they already have pipelines available for AMR research, 16S, SARS-CoV-2 analysis as well. So there's a lot of resources available from the Oxford Nanopore community as well. You can do guided analysis, which is basically trying to teach you how to enable yourself to do bioinformatic work as well to set up your own pipelines. And then if you are a bioinformatician by trade, you can also use the GitHub page to do custom workflows as well. So I truly hope that this quick talk gave you an idea of what the technology can do and give you an idea on how to ask bolder questions within your research and how to make no compromises with your sequencing data. Then also just, we do have an online web shop, so if you want more information on anything, you're more than welcome to stop by the stand and speak to me today. And you're also welcome to view our online shop to do instant quotes, check our stock availability, and see our pricing online as well. Thank you so much.
Thank you very much, Letitia. Letitia is available outside, so if you've got any questions that you would like to ask her, please visit stand. Thank you, Letitia.
Thank you so much.
As this session comes to a close, I would like us to thank the speakers with a round of applause. Lastly, I want to apologize for taking 10 minutes of your tea time. I hope you'll make the time afterwards. Otherwise, this is the end of the session. Thank you very much for being with us this morning, and enjoy the coffee outside.
I'm with my colleague, Dr. Mohali here. We are chairing this session for you. And we are thrilled to have you here. And I want to enlighten everyone here that from looking at the program, I see a very exciting lineup of presentations. So I would like everyone to sit and relax and wait for those brilliant presentations from our student. So without any waste of time, I would like to call upon my colleague here to come and introduce our guest speaker, Professor Jones Ngambi.
Thank you, Dr. Munyama. Oh, there's no balance. Thank you so much. Mine is just to introduce Prof. Ngambi. Prof. Ngambi Jones is affiliated with the University of South Africa under the Department of Agriculture and Animal Health. He has authored 128 publications with 70,000, listen carefully. Yeah, I'm summarized here, but it's around 70,000 reads with around 1,600 citations. His current research insight include improving productivity of indigenous livestock, including chickens and goats, through efficient nutrition and breeding, and also manipulating nutrition to reduce methane production and emission in ruminants. He employs methods such as ration manipulation and breeding. Currently, he is working on the utilization of taniferous feeds for reducing methane production and emission in goats and cattle. Prof. Ngambi, the floor is yours. Colleagues, let's give him.
What do we do here? Yeah. Did you provide it? Yes, it was there. Thank you very much for the introduction, and you are welcome to listen to whatever I have. I think it's important to emphasize right at the beginning that there isn't enough time for me to go into details of what I'm supposed to talk about. So when you have a question, then we can go further into that particular aspect, and I'm here to answer whatever is possible. So I'm going to talk a bit on funding opportunities for postgraduate students and the importance of publications. I think much of it possibly will dwell on my experience over a lot of years. Yeah. So the first thing you have to do is to identify the agencies that are involved in funding. I think it's very important that as a postgraduate student, you have to have some funding, basically for research, also for your upkeep. And I should say right at the beginning that I think I've been to many countries in the world, and South Africa at the moment is the best for opportunities for postgraduate students in terms of funding. Some people may argue against it, but if you go to other countries, you don't have those opportunities. Well, that's why I've put South Africa at the middle. I'm going to go over it, but I'll put some details later in terms of South Africa. So in South Africa, you have NRF, Water Research Commission, IDC, provincial governments, etc., which are sponsoring postgraduate students. But the main one is NRF, because this is a government agency which is mandated to do that particular job. In terms of Europe, is possibly the second one in terms of sponsoring postgraduate students in South Africa. And the Commonwealth at the moment is almost at the top. But you have other agencies like the European Commission, Union, FLIA, this is a Belgian funding agency, etc. And I am happy to inform you that I've gone through all those. In terms of funding, I was funded by Commonwealth for my master's degree at the University of Aberdeen in Scotland. I was funded by Commonwealth for my PhD at the University of London. But I've also worked closely with a number of funding institutions in Europe. For example, the EU. I had an EU project involving 16 African countries and 12 European countries. At that time, I was representing Lesotho. I was in Lesotho, at the University of Lesotho. So I was the representative of that country. I've also been involved in FLIA, which is a Belgian funding agency. This funds a lot of projects in the world. I was leading the project on feeding, on feeds and etc. And let me just give you a perspective of it. It was a project for 10 years. They sponsored 30 PhD students during that time and over 100 MAC students over that 10-year period. I'm talking of the project that I was leading, but there were other projects in that big project. So, yeah, these foreign projects involve a lot of funding for students. My belief is that a number of institutions have these projects. If we don't have, then we have to look for one, and it's up to us to get it. There is also, you have a lot of agencies in North America, USAID. I was involved in a USAID project for 10 years. And that project involved 25 African universities and 5 USA universities. And it sponsored a lot of students during that particular time. It was based at the University of Makerere. I think in South Africa there were 2 universities involved, my old university at University of Limpopo and Pretoria University. So they do sponsor a lot of people. In terms of other countries, I think the Chinese agencies come first there. At the moment, there are quite a number of people sponsored by Chinese agencies. And I think we have some who have gone through that agency in this room. I'm not mentioning them, but we have. But at the end of the day, what matters is really the human resource development. Human resource development. All these are aimed at developing the human resources in terms of knowledge, in terms of capacity, in terms of understanding the situation in a particular environment. Why? My friend, it's not moving. Why is it not? Ah, so it has to be this side. Oh, it's the laptop, okay. Yeah, thank you. Huh? It's not. Oh, this way, okay. All right, I said I'll come back to NRF. And really, NRF is the largest agencies in terms, largest agents in terms of sponsoring postgraduate students. I've just given this just some understanding of what is happening at the moment. For example, you have the honors students, and it is open up to November. And then you have first-time master's students. It opened on 23rd May up to 22nd November. First-time doctorate students, unfortunately, it closed on 1st July. And master's and doctoral extension support, it is still open. But I think it's important to remember that this is a website you should first visit. And you should be visiting this website regularly, because there are a lot of opportunities. In terms of foreign agencies, many of them go through NRF. So they will be attached to NRF. So it's better to be visiting that particular agency. But also remember that NRF funded projects include a lot of scholarships for the students. I'm sure there are quite a number of projects in this university sponsored by NRF. I'm not very old in the university, so I don't know much, but I suppose they are. So there, the students also can be attached to the project. Just one point to remember is that there's this global fellowship for moving to other institutions outside South Africa. I think we should take note of that one. If you have an NRF sponsorship instead of, in terms of doctoral or postdoctoral, go and apply for that. You spend some few months at another university outside this country, and you learn a lot in terms of knowledge. I hope it will work. Why? Oh, so now it is working faster. Okay. Okay, yeah. In terms of foreign scholarships, I'll spend a bit of time on this one. possibly the most crucial one at the moment is the Commonwealth Scholarship. And it is opening somewhere around October. So if you are looking for a scholarship, you're doing a master's, you want to do PhD, please visit that website. The good thing with this scholarship, and I'm talking from experience because they are the ones who sponsored me for my postgraduate studies, they pay for you, they pay for your research, they also pay for your spouse, they also pay for your children. So you gain a lot in terms of that. And if you are already working, you don't need that money from your home to live that side. So it involves a lot of paperwork. But because I've gone through it, I've helped a lot of students, particularly from the University of Limpopo. I'll give you one example. Possibly some of you know her. I supervised for her master's. And a few years ago, we looked into this, and she applied for that, and she got the scholarship to go and study at my former university, University of Aberdeen, for her PhD. Why I'm mentioning this, before she could even finish her PhD, when she had just submitted a thesis, she was grabbed by a very famous university in US. I'm not going to mention the university because then you'll find out. Well, just to cut the story short, she's now teaching at a university in USA. So take note of that, and you can do it. Yeah. Okay, as I said, also the foreign projects normally involve a lot of sponsorships. Unfortunately, these are only open to students attached to the projects themselves. So, but one should look for this. If this university, if we don't have a huge one, we should start thinking of getting one, because that will put the university up there in terms of ranking. I hope not. No, it should work. My friend, I don't know. Or can I use? Why is it not? Huh? What? Yeah, I'm going. Should I go? No, go back. Okay, go next. Yeah, that's the one I was using. So you're putting it up here. Thank you. Sorry. The next thing we can look at is identifying where to publish. So we are talking about publication. I think it is very important to remember that the Department of Higher Education and Training has a list of journals where you can publish. This is important because they pay money for publication in this country. And the institutions which publish a lot, they get a lot of money. I'm not sure now, but the last time I checked, I think one publication was something like 130,000 rands. All I'm very sure of is that if you publish and you are alone as an author, you get around 40,000 at the University of Limpopo. I'm not sure here. That's not a small amount of money. So, but the idea behind it is that they want to increase knowledge, knowledge generation in this country. And I think they're doing a very good job. So you look at NRF accredited journals or higher education accredited journals and look at where you can publish. The second one is reputable journals in your field. So look at the journals that are very reputable in your field, because that matters a lot. In terms of journals with high impact factors, I think you published one in that one. I think so, in Nature. But it's not easy to publish in Nature. No, it's not easy. So look at the impact factors and see where you can manage. But don't take the ones with 0.001. Take the Nature. I think has got somewhere around 65 or so. Now I'm not sure how much. How much? You can't remember. Okay. But the problem is that sometimes it's very difficult to publish in such journals, and you may not be able to do that. And normally, for example, Nature, you have to involve a lot of other researchers finding out information over a period of time, and etc. But it's important to remember that. Publication time, there is short time versus long time. Some journals take more than 1 year after submission before it can be published. Others take shorter times, maybe 3 months or 4 months. So look at it and see where you can publish. Publication costs, that is very important. A number of journals now charge money, particularly those in terms of the open access, because they don't get money elsewhere. So check with the university how much they can afford to pay. I'm not sure how much it is here, but I know my old university was 30,000 rand. That was the maximum they could afford. But there's another way you can go around it. If you have a project, you can get that 30,000 and then get some more money from your project. So it's possible. But again, you find that some journals charge a lot of money. The average is possibly 1,000 euros, but you get some up to 2,000 euros. And once you start going up there, then you really have to think about it. Publication type is open or the traditional journal. The open access journal now is becoming very common because people can access it and they can get information. But remember, all you want is to make sure that your findings are taken up by other people. Language and style, yes, it's very important to know that wherever you publish, they have a particular style, and don't go over it. It's important, otherwise they will not take your manuscript. I mean, possibly the majority of us may be possible to publish in English, but if you can publish in French, then you choose a French journal. If you can publish in Afrikaner, then you choose an Afrikaner journal. So you have that idea. Then you have the local versus international. Both are important. To me, both are important, depending upon what you are working on. I know there are some people who feel it's better to publish in international journals. But, I mean, it's up to you. However, as long as that journal is accepted by NRF, that's fine. And for rating purposes, it's important to choose journals which are reputable, are very reputable, rather than journals which are not. Research impact, I think this is important. If you are working on vendor chickens, I've been working on vendor chickens for years. That's what has come to me. Really, it might be very important to publish locally here, and so that people can benefit from it. But also you can publish wherever. It doesn't matter. I hope now it will work. Why? Huh? Down. Sorry. It looked at you and said, okay, he's coming. Do it. Okay, let's look at why publications are important. I'm not going to waste a lot of time. I think One is career advancement. It's important that you go ahead in that area and you advance. Second is professional recognition. If you are working on nutrition and you're publishing a lot, then you are recognized, and then therefore you can have funds and etc. Opportunities for collaboration. I will tell you one thing. For example, I said I was involved in the USA project. It was called RAN, and it involved a lot of universities in Africa and United States. I didn't apply for that. They actually approached me. So once you publish in certain areas, other collaborations come up, and they will approach you. I don't know. Huh? It's it. Okay, sorry. Thank you. Sorry. The next one is increased visibility. I think once you publish, then, as I said, then people will know that there is someone who's working on this. And then you can have collaborations, you can have funding, and etc. Impact on society. I think we all aim at finding new knowledge. And we expect that knowledge to have impact on society. These days, you are not going to have, like the old days, one person working on his own and finding out, no way, that is gone. So your research will have a small contribution. You may not even notice it later. But when everything is put in a holistic manner, your part is played nicely. So it will have some impact, knowledge in terms of credibility and trust. Once you publish, people read and they start trusting you, and they can give you money. Yeah. No, now let me try first. Either the batteries are flat, or can you keep it? Yeah, and then, yeah, that's better. Why publication is important, continued. 7, professional development. You know, you publish so that you develop your professional career in that particular area. Inspiration for future research, that is very important. People don't realize it, that we always look at someone who has done work in your area and say, I like that. That inspiration comes out of publication. Then we have contribution to the field. Of course, the knowledge will contribute to the field. But I think at the end of the day, the most important thing for many of us is possibly financial benefit. And as I said, in this country, if you publish and you're at an institution, you get money. The institution gets money. So the more papers we publish, the more money comes to our institution. But also the more money comes to your pocket. I remember I published, the first time some students published with me, at the end of the year, when they got the money, they were so excited. And they started working very hard to publish more. So, yeah, there's money there. People will say, no, don't do research because of money. But the truth is, we can't go away from there, no. Next one. Okay, some important points to note about publishing. I think, remember that your manuscript is aimed at finding new information, but also at solving some problems. And therefore, whatever you do in that research is aimed at adding knowledge. And you should make sure that that is very clear in your manuscript, the knowledge that you have added. And that's what they look for. Has this added some knowledge? If yes, they'll publish it. Remember also to use research tools. There are quite a number of research tools that you can use to get information, knowledge. For example, the Google Scholar, Scopus, Microsoft Academic. There are many of them. I think we always use them. But remember, if you want to get latest information, use them so that you can know what is happening in your area. Plagiarism and similarities, that is very important. I want people to note that when you send your paper for publishing, they check it for similarities. And if there are so many of them, they will return it. So don't wait until they bring it back to you. Check it and change accordingly. I think those of us who have been publishing for some time know how to manipulate that. In other words, don't take the statement that someone wrote. You can have the same meaning, but in a different way, different words. That's what they want. They don't want you to copy that particular statement. So that is important. But again, this comes out of the skills development and knowledge. I'll spend a minute or so on peer review. You see, when you send your paper for publication, they send it to a person in that particular field. And peer review basically is a process by which manuscripts are scrutinized by other researchers to ensure that only the best quality manuscripts are published and provide constructive feedback. I wanted to talk about constructive feedback. The first time I published a paper from my MSc dissertation was some years ago. I was at the University of Aberdeen. I think it was red. It came out red. But my supervisor said, you know, what you should get out of this is the constructive criticism. Look at it and improve on it. And I have kept that in mind all the time. And once you improve it, you know, if that person even rejected it, but that person has given you some idea of how to improve your paper. And therefore, you improve it. If they don't want it there, send it to another journal. That is very important in terms of publication, particularly for those young ones who are coming into the system. You have to follow the author guidelines. They are different, very different. So read some other articles in that journal and see how they write their manuscripts. Remember, knowledge also involves copying ideas from other people. The next one. Okay, so in conclusion, I want to emphasize 2 points. That there are a lot of opportunities for postgraduates in South Africa, both in South Africa as well as abroad. Make use of those opportunities. Publishing your research work is very important. To me, it is important for your development as well as for your financial benefits. So it's important for developmental and financial benefit. Go to the next one. Yeah, thank you.
Thank you so much, Prof. Ngambi, for such an informative session. I've noted down some of the inspirational tips around funding applications, the importance of publications, and also taking advantage of the international opportunities. Thank you so much, Prof. This will really help our colleagues and also our postgraduate students. And we are also very grateful for you and for putting together this work. I think it will really benefit us. And as a distinguished professor, we normally consult you. I'm already in that journey, so we are really appreciative of the work that you are doing in this space of research and ensuring that our researchers are capacitated and they are helped and always supported. Colleagues, we will have 5 minutes for questions. We will take some few questions for Prof. Ngambi.
Thank you. Thank you, Prof, for your interesting presentation. Mine is not really a question, just maybe to emphasize, some of the aspects that you were raising in your presentation. I think the issue of funding is quite crucial, and I feel this talk is quite relevant for our students, because what we have noted at the college, or rather at the science campus, is that our students are not taking advantage of the grant opportunities that are available. And I think your talk is quite relevant, Prof. Maybe they will have enough energy to go and look at those different grants. And then the second thing is more on issues of publication. You talked about how one should perceive feedback. When we go for presentations, when our students are presenting in different research groups, you have students that are not able to take criticism. And as you've indicated, it is quite crucial because it serves as a learning curve. And I think that not only goes for our students, also for the researchers, because when you publish, you get your paper with those comments. Sometimes you think they're nasty, but I think it's important to really appreciate them. And thank you for re-emphasizing that, Prof. Thank you.
Thank you.
Thank you, Prof, for that wonderful presentation. Prof, I just want your take or a comment on, there's talks on the street, but it's not really confirmed. You have a link of sort of a good impact factor, but there's a publication house, in this case, MDPI. So what is your advice to colleagues and students? Because you also touched on something, the publication time. Does the publication time relate to quality? Because there are houses that takes forever to publish, and there's houses that take few weeks for you to get a clear picture. So I just want you to give a clarity on that. Thank you.
Yeah, thank you. The first one is very important that you look at the list of publications emphasized by the higher education and training. Yes, the particular publisher you mentioned, I'm not going to mention. Yeah, there was a time, I think a year or so ago, when they were actually removed from the list. And then they were put back. I think avoid those. But there are certain areas where they're very good. And it might be important to publish in those areas. Yeah. The other one was, what was it?
Just the correlation between time.
Okay, the time, yeah. Well, not really. I'll give you one example. I publish a lot in the British Tropical Animal Husbandry and Health. They take at most possibly 5 months, and they're very good at it. However, there are certain journals which take longer than that. So, of course, if a journal is publishing your work in 1 month or 2 months, something's wrong there, because it takes a lot of time for the reviewers to go through, and you don't expect someone to go through within a very short time. So, yeah, you see, in terms of the journals, you have to think ahead. Don't publish in a journal that is on the list, but is questionable. However, there are certain fields where you have to publish in those journals. You can't go, you can't run away from there. Yeah, thank you. I hope I've answered.
Thank you, Prof, for such an informative presentation. I just want to ask, you mentioned that sometimes we do get invitations from other academic institutions, and that also sometimes happens with some research, journals, where they will invite you to present your work with. I just want to find out from Prof, what would be your advice in distinguishing between the fake invitations and the genuine one? If you have some tips for us, I appreciate it. Thank you.
Yeah, thank you very much. Yes, I get those almost every day. And the first thing you do, if such a journal asks you for doing that, check if that journal is on the list from the higher education. I think if it is there, then you don't have to question it very much. However, in so many cases, they just want to publish, and they want materials. And you have to be careful, yes. Thank you. Sorry, you haven't, yeah, just, yeah, just, I can hear you.
Okay, all right. Sometimes you get invitation where they say you're invited to be a speaker at such an event or conference.
Yeah. Okay. Oh yeah, sorry, I missed that one. Yeah, no, that is very important. I normally get invited to present something or to be a guest speaker. Attend those, regardless of. I mean, your paper may not bring in money, but when you are there, you expose yourself to other collaborations and etc. I do that. Yeah, particularly at the conferences. It's very important. But if a university invites you to do that, run for it. Don't even walk, you run. Thank you.
Okay, thank you. Maybe just one here.
Yeah.
Okay, as a very young fellow, who is still looking to publish, I noted something where you mentioned that we shouldn't target journals that have too high impact factor. I'm wondering what will be the disadvantage if you have the resources to publish there?
No, maybe I might have misled you. What I meant was that, if you are, for example, I didn't give the example I gave. If you have written a paper yourself and you want to publish it in Nature, they're not going to accept it because you haven't collected enough information. And they want that information to come from different areas. They want information on that particular to be at the top. So, you can try it and get the rejection, but aim a little bit high, but not very high to start with. A good example, as I've said, how many were you in that publication? Is it 60? 6. And not from the same area, from different institutions. I have seen a paper at the University of Cape Town in Nature. There were 100 and something authors involving almost all the continents on that particular aspect. So, yeah, I'm just saying, yeah, be careful about aiming very high. Yeah, thank you.
So more a comment than a question, but I think the question that was asked from this side of the audience about when you get an email that invites you to be a keynote speaker, for you, that's a real email and you're a real keynote speaker. For a junior staff member, that's spam. And because, last week I was asked to be a guest editor on a journal, and a reputable journal, on a topic that I personally wrote back to them and said, I am not an expert in this field, and I'm completely inappropriate, and I don't know why you think I would be suitable. And they wrote back and said, thank you very much. I didn't know what to say. And then a week later, I got another one from another journal with an equally inappropriate question. But what you see, and we have seen a lot less of it in recent years, is that certain countries, which I shall not name, but you can imagine, are building an industry of conferences, and they invite you to come. They don't tell you as a keynote speaker, they'll pay your fare. When you've accepted and you've sent your abstract, then they come back and say, well, of course, you've got to pay for this and that and the other and whatever. And then when you get there, you discover that, you know, it wasn't.
There's nothing,
So real conferences are real. And they have a track record, and they're known in your field. I mean, in my field, plant and animal genome, which is now not just in San Diego in January, but there's one in Asia, and there's September, there's one in Australia now. We know that's a real heavy-duty conference. And, you know, I can guarantee I'll never be invited to be a keynote speaker there, because their keynote speakers are megastars. So there's an element of saying, if somebody writes you an email like that, take it to somebody more senior and say, do you think this is real? And they're going to say, nah, don't even look at it. Because you're not going to get value from it. You're going to get value from a top-level, reputable conference. And don't be tempted by the idea that somebody fished you out of a database and thought you were a superstar, because I'm sorry to say, you weren't. I think to answer Lucky's question, and Lucky and I are very much involved in the same paper, this question of publishing in Nature, or let's say more broadly, the Nature group journals, because there are 50 Nature group journals together with the Frontiers journals and the Nature journals. Nature's very special in the sense that they want to publish stuff that is, firstly, of general interest, and secondly, of high scientific value. And that is a very difficult combination to get. But when you get it, you can publish in Nature. But it's very rare for anybody in South Africa to publish in Nature. The Nature group journals, Nature Genetics, Nature Biotechnology, Nature Plant Science, are high-quality, high-impact journals which you can get into, but you've got to have not just good data, but a good story. You've got to have a good general interest thing as well as having high-quality science. And Lucky's Nguni sheet paper, he hopes it's going to make it in. There's a bit of a fiddle going on in the background with the editors. So I think he will get in. But it's not coming out of nowhere. It's coming out of a relationship with the journal. And that's what's going to make that access a bit easier. But these are all complicating factors. And yes, I think to answer another question, your journal has to be in DHET. In fact, I was saying to Olivia the other day that the college shouldn't recognize any publication that's not in DHET, because you don't get subsidy for it. And so the college should say you can't have it on your performance agreement. Non-DHET journals, you'll not get anything on your performance, and that will stop it. Because if it's not in the DHET list, there's no subsidy to the university. And if it's not in Scopus, which is a validated list of 28,000 journals, of which I saw a paper the other day said, I think 384 were dodgy, which probably means they've gone already. But if it's not in Scopus and it's not in DHET, don't even think about it. You know, don't be tempted by a low-impact journal, because it is of no value career-wise. And when people like me read your CV, they're going to say, but what are you publishing this rubbish for? Okay? I've published, you guys looked me up on Google Scholar, which I don't use. I've published 84 papers in my career. Every single one of them is in Scopus. And the other stuff is all sorts of nonsense rubbish that finishes up in Google and you can't get rid of. But it's not publications. It's the conference abstracts and, you know, silly stuff. So Scopus is critical to your self-criticism of your publication record. The citations there have credibility. So by reference, I have 5,500 citations in Scopus. I have 9,000 or plus in Google Scholar. But I don't think the Google Scholar number is a useful number. Yeah, it's nice to think 9,000, but it's not true. It's 5,500.
Let me just interrupt, Prof. Thank you so much. Thank you so much for a detailed comment. Colleagues, this conversation can be continued. Dr. Andrianse is with us, so stay tuned. After lunch, we are going to continue with this conversation, and she will clear some of the questions that you are having, because she's a specialist in that area as our librarian. And I will hand over to my colleague.
Okay, thank you. Colleagues, now we're moving on to our very important people here, our students' presentations. Just to note down and to remind everyone that the importance of Symposium, actually, we are here to actually learn and share and again to grow together. And then students, I urge you at least to make this experience very memorable. And then I'm going to call upon our first speaker by the name of Ms. Penny Makumbila. Okay, just to note a few, in every house we have rules. As student, you only have 15 minutes, which means you have 10 minutes for presentation and 5 minutes for questions. Thank you.
I think it's 15 for your presentation. Can you use this?
Good day, everyone. My name is Penny Makumbila, and I am a PhD student under the supervision of Dr. Figlin, Dr. Rawani, and Dr. Muedi. So today I will be presenting on transcriptome analysis of common bean, that is Phaseolus vulgaris, in response to rust, that is Euromyces appendiculatus. The presentation will delve into the background of common bean, and I will move over to the materials and methods that were used in the study, and then the results, and then the discussion, and then after that, the conclusions before the acknowledgements. Well, common bean is highly impacted by the occurrence of rust that is caused by the detrimental fungal pathogen Euromyces appendiculatus. And what the pathogen does is it forms yellowish to whitish spots on the other side of the common bean leaf that eventually turn brown, and consequently resulting in the leaves having hollow brown spots that is inhibiting the occurrence of photosynthesis and eventually impacting the pod sizes and the yield. The management of the pathogen has mainly been through the use of biological methods, such as the use of Trichoderma species and Pontua agglomerans. Cultural practices such as deep cultivation, adjusting your planting date, crop rotation, and minimum tillage. And in figure 2, suppose you were to use or to have a crop rotation program for 4 years that rotates with different species, then what it would mean is you would not be able to get, the nutritional benefits of having common bean, meaning that in a 4-year cycle, you only have one season of common bean, and that is not very ideal. And chemical control has been done through a combination of difluconazole and azostrobin. And with chemical use, the problem is that the pathogen is constantly evolving and has thus had it has systemic resistance to a lot of chemicals. And therefore, the use of resistant genotypes promises a more viable avenue that we should explore in cropping or farming in common bean, which is why we conducted this study. We wanted to evaluate the response of Phaseolus vulgaris to a virulent strain of U. appendiculatus, that is race 1, at different time points that are quite critical in plant development. And we used an RNA sequencing approach in doing this. We utilized 2 genotypes in this study, that is Golden Gate Works and also Tibas RR1. These genotypes were obtained from the Agricultural Research Council, and what we did prior to planting, we surface sterilized the seed in a 50% bleach solution, and then the spore concentration was adjusted prior to spraying the underside of the leaves using a spray gun, and sampling was conducted at 14 days post-infection and 21 days post-infection. And then during sampling, these samples were snap frozen in liquid nitrogen and then stored in a -80 ultra freezer prior to RNA sequencing analysis. The RNA sequencing was conducted at ARC, that is the biotechnology platform, where an MGI instrument was used. In the data analysis pipeline, after receiving the data from ARC, the data underwent a quality check where we used trim galore and then aligned to the genome in high set and then sorted the reads in some tools and then string tie, and then we used DE-seq2 to get the differentially expressed genes. And commonly in the analysis of transcriptomic data, there isn't, an analysis of potential biomarker discovery. So in this study, we further took the data or analyzed the data in Simca sartorius to sort of, get potential biomarkers that could be used in this sense. And then furthermore, we took the DEGs and we wanted to look at the functional categories that could be responsible for tolerance to the pathogen. During the phenotypic evaluation of the study, Golden Gate Works, according to the severity score, exhibited severe symptoms of the pathogen, and that is at 14 days, as you can observe in figure 5A, and at 21 days, the secondary leaves were already showing signs of infection. Meanwhile, on the underside leaves, that was Tibas RR1, and at both time points, there were no symptoms of the pathogen. And from evaluating or mapping the transcriptome, Golden Gate Works expressed over 2.5 differentially expressed genes. And at first, I panicked because I was like, okay, 2,500 genes, differentially expressed genes for Golden Gate. Meanwhile, Tibas RR1 at 14 days only had, I think, about over 200 DEGs. And it was the fact that the data was getting a lot more interesting. And from observing the volcano plots, you can see that literally at 14 days, that is figure 6A, at 14 days in Golden Gate Works, there were very few genes that were not differentially expressed. The rest of the genes were up and down regulated. And at 21 days, a lot of these genes were not differentially expressed. Meanwhile, if you observe on the other side, that is figure 6C and D, Tibas RR1 sort of had a more consistent regulation or expression of genes. The genes that were expressed in both genotypes at 14 days post-infection included heat shock proteins, cytochrome monoxygenases, and terpene synthase, among others. And at 21 days, we observed a peptide that enhances cell death and the WRKY transcription factor. And zooming into these results, we can observe that, for example, the cytochrome gene CYP 78C4 was downregulated in both genotypes. And this downregulation has been found to be associated with linalool and terpenoid metabolism. And on the other hand, at terpene synthase 21 was upregulated in Golden Gate works at 14 days. And in a study that was evaluating terpene synthase 21 has been found to be associated with increased levels of methyl jasmonate, and those increased levels of methyl jasmonate are known to promote, to accelerate defoliation, and they do not promote plant growth, which possibly could explain why, after evaluation at 14 days, the primary leaves of Golden Gate works had fallen off. And another interesting gene was the WRKY transcription factor 70, which was again upregulated in Golden Gate, while downregulated in TBUS RR1. In a study that was conducted evaluating the response of chickpea to Fusarium oxysporum, WRKY transcription, WRKY 70 was associated with genotype susceptibility. In looking at the potential biomarkers we could use for tolerance to the pathogen, NBARC domains were found to be part of the biomarkers, together with thiolase protein families, among others. And zooming into the expression or potential biomarker pattern expression, as you can observe that gene ABO4 was exponentially expressed in the genotype Golden Gate works at 21 days. I think it was expressed at over 20 log 2-fold change. And this overexpression of the gene has been found to be associated with flowering. And at 21 days, it could mean that the plant was already going into flowering mode, possibly caused by the pathogen. And it would possibly lead to, flower abortion, which would impact the yield. The functional categories included molecular functions, biological processes, and KEGG pathways, and these included protein serine activity at 14 days in Golden Gate, and at 21 days, opoplast was enriched. And in TBUS RR1 at 14 days, despite expressing, fewer differential genes, there were a number of enriched functions that were responsible for stress response. And at 21 days, again, there was increased chlorophyll and photosynthetic activity. We have submitted this manuscript in a journal, and the paper explains fully, the functional categories and what genes were responsible or were backbones to these functional categories. So we are hoping that it will be published soon. And yes, in conclusion, the study provides a basis for plant breeders, as evaluating of genotypes at critical stages is very important. And gene expression, the gene expression patterns that were found in this study can be used as a guideline for selecting superior genotypes. And an integration of biomarker discovery into transcriptome data would be very beneficial, as pipelines do not include that. It's mostly this was differentially expressed and that is it. There's no way of, selecting biomarkers. And if given funds, it would be quite beneficial to actually, look into the genome of the pathogen itself to see how it interacts with common bean and also evaluate a lot more genotypes and other races of the pathogen because there are over 90 races of this pathogen. And then I would like to thank my supervisors for their constant support and the ARC, and the NRF, and of course the University of South Africa for the funding and also, the library services that is provided by the university. It's been really great. Thank you very much.
Thank you, Ms. Makumbela, for the very interesting and informative presentation. Colleagues, it's time for questions. Any questions, comment?
Controls. Tell me, where was your day zero?
Pardon?
Where was your day zero data?
My day zero, the control experiment.
Mm-hmm.
Okay, the control experiment was only mock inoculated with distilled water, and it was put through like similar conditions to the race 1 infected plants, but it was just distilled water the whole way. So yes, there was a control.
Was there a day zero control? As in, at the very beginning of the experiment?
Zero as in nothing was done to the...
On day zero. At the beginning of the experiment, did you do RNA-seq on those samples?
No, I didn't.
Okay,
it was only at 14 and 21
okay,
days.
and how many replicates?
I had 3 replicates.
And how many reads in the sequencing?
How many reads? It was millions of...
Yeah, I know. How many millions?
I off the top of my head, I cannot remember, but it was quite a lot of reads because I received like, I think it was 240 gigs of data. So it was a lot, yes, but it was millions of reads.
Okay. My point being that you must describe the statistical power of your experiment in order that I can make sense of it sitting in the audience. And I'm a nice guy. But you've got to describe the statistical power, which means you need to describe the controls properly, and you need to describe how much data you've worked with, because those are key to understanding whether 2,500 genes being differentially expressed makes sense, which is a very big number.
Yeah, it is.
And the question is, are you looking at developmental changes, or are you looking at infection changes? And that's why I'm asking what the controls are and what you're comparing with. Thanks.
Thank you. Thank you very much for that.
Any other questions, colleagues? Comments?
Okay, I just have one. I noticed you did mention that you did validation with qPCR. I might have missed it. I didn't see the results from your validation.
Oh, yes, I did validation with qPCR for both the differentially expressed genes and also the potential biomarkers. I just did not include the results here, but they did confirm our findings, which is why I said the paper is a lot more explanatory because initially I think I had over 32 slides, and that would have taken up most of your day. So, and the data will be publicly available, I think, once we publish. I think I've set it to release the data in 2 months. So, yeah, we did qPCR at Ingaba, and yeah.
Okay, thank you, Ms. Makumbela. Is there any other question? No, I think we're fine. Oh, okay. Okay, let's give him a chance.
Thanks, Penny, for that wonderful presentation. Mine is just an agronomic concept because in the whole experiment, there was nothing about the management of non-experimental variables, so that maybe the expression could, from where I'm sitting as in an agronomy space, is the expression maybe a function that also the crops themselves were not given nutrition because there's no much on the management part of the whole the experiment, the non-experimental variables. And the second one will be the expression of, the expression of the gene, of the, is it parasite, the virus? Is it based on the conditions that prevails? Maybe the humidity and other things? Will if that's the case, do you think then the cultivars that you found to be more resistant, will they do the same under different agronomic zones? Thank you.
Thank you for that question. With regards to the agronomic aspects, we kept everything optimal. We used oxidic soils, and then we fertilized, and we kept everything else optimal, but the main focus was the pathogen itself. And yes, the pathogen thrives under highly humid conditions. There's been reports of the pathogen is highly prevalent in KwaZulu-Natal and in the northwest in areas that produce common beans. So it requires humidity to actually thrive. I'm not sure what your other question was again. Yeah, which is why we kept everything optimal in the sense that gene expression is quite tricky. If possibly I had evaluated the genotypes and like outside, the gene expression patterns would have been something else. But we kept the experiment separated from each other in different compartments so that everything else is not a factor, but the only factor was the pathogen. So yeah, I hope that answers your question.
Okay, colleagues, thank you, Penny. Thank you for the nice presentation. I think a round of applause, please. Now we're moving forward to our next speaker by the name of Mr. Makwarela Tsirelezo. The floor is yours.
You need to adjust your mic.
Good afternoon, everyone. As already introduced by the co-chair, I'm Makurela Tsaredzo, and it's pleased to be standing before you guys to present this topic of great honor, or of good significance, especially in the agricultural sector. So my topic for today is diversity and distribution of hard ticks infesting cattle in South Africa. I believe most of you have got preferences when it comes to red meat, whether in which store you would purchase it at. It may be a butchery or it may be a retail market. But do you know that the infestation of ticks or the presence of ticks in our livestock have got so much influence on the quality of meat you get, to an extent that if there are heavy infestations, it means that the meat can also not be available at the market. But since most of you might not know, let me educate you. Okay, what are ticks? Ticks are ectoparasites that carries pathogens, such as your bacteria, viruses, and protozoan. These pathogens are able to cause diseases in humans and animals. And which diseases are those? It includes diseases such as your Lyme diseases, tick paralysis, anaplasmosis, and babiosis. There's about 923 tick species that have been recorded globally, of which 700 of them belongs to the hard ticks and 200 of them belongs to the soft ticks. And then in South Africa, we have one additional family of ticks that shares both characteristics of both soft ticks and hard ticks. Then you would ask, TG, why are you doing this study? What is the significance and why is it important? To study the tick diversity and the distribution is very crucial to develop effective strategies in order to control them. So we've got encountered, or there's been an estimation of direct and indirect losses that are caused by tick and tick-borne diseases. About 22 billion to 30 billion has been reported to be spent on tick and tick-borne diseases, of which in South Africa, about 33 million sorry, if I'm excused, Doctor, I don't think this is the right presentation. Sorry about that. Can I bring my machine?
Yeah, you can bring.
Okay. Colleagues, just bear with us. There's a technical error here that still trying to fix. 2 minutes, please. Okay, colleagues, just to save time, Mr. Makwarela will come next after our next speaker. I think Mr. Lazarus Katlego Morani is ready for us. The floor is yours, Mr. Morani.
I'm hoping it won't be another issue. It's fine, I'll just try. Oh yeah, technical error here, colleagues. It's so unfortunate, but I trust my technicians. Let's call another speaker by Miss Celia Mzeza. Do we have her in here? Celia Mzeza. Perfect.
Just use the keys or the space bar. Which ones are the keys? Here. Okay. I'm gonna use left and right. I just want to say something.
Good afternoon, ladies and gentlemen. My name is Celia Muzeza. I'm a PhD candidate in the Institute for Nanotechnology and Water Sustainability. Today, I will give a talk, which is part of my PhD work, entitled The Occurrence of Priority Phthalate Esters and Potential Health Risks of Sewage Sludge Application to Soil for Agriculture in Gauteng, South Africa. On the screen is the outline of my presentation. Sewage sludge, the inevitable byproduct of municipal wastewater treatment operation, is a critical issue globally due to its increasing volume and impacts associated with its disposal. Amongst its disposal options is the utilization in agriculture, because sewage sludge is a good source of organic matter and mineral nutrients. However, the application of sewage sludge to agricultural soils may introduce a wide range of pollutants, including contaminants of emerging concern, such as phthalates. Phthalates are an abundant and essential class of synthetic organic compounds that are variously classified, including into priority phthalates, and are widely used as additives, solvents, and plasticizers in numerous everyday products, including plastic packaging, personal care products, pesticides, and oils. Priority phthalates are so named due to the adverse effects that they have on the environment and humans. However, when phthalates are added to products, they are not covalently bound to these compounds, and so they can easily leach into the products and eventually into the environment, causing numerous effects such as metabolic disorders, cancers, reproductive disorders, just to mention but a few. Globally, wastewater treatment plants have been singled out as a primary source of these priority phthalates to the environment. This is because wastewater treatment plants cannot effectively remove the pollutants, or rather the priority phthalates, from the wastewater. And so they are discharged either together with the effluent or with the sludge into the environment. South Africa specifically produces more than 944,821 tons of sludge per annum. And a significant percentage is utilized in commercial agriculture, which occupies about 1.3 million hectares of land and contributes about 25 to 30% of the country's agricultural output. Gauteng province specifically produces more tons of sludge per day compared to the other provinces in the country. As such, our team set out to investigate the occurrence of these priority phthalates and potential human health risks that are posed by the application of this sewage sludge to soils in agriculture. The objectives of the study were: 1, to characterize the sludge which is produced from wastewater treatment plants in Gauteng; 2, to determine the concentration of 4 priority phthalates, which are benzoyl-butyl phthalate, diethyl phthalate, dibutyl phthalate, and di-2-ethyl hexyl phthalate in the sludge; and finally, to evaluate possible human health risks that could arise from the application of the sludge in agriculture. In order to achieve these objectives, the study was conducted in 3 wastewater treatment plants, as shown on the slide: wastewater treatment plant 1, 2, and 3, specifically in the Joburg municipality. And notably, all these wastewater treatment plants, they utilize the activated sludge technology to treat wastewater. They treat domestic and industrial wastewater, but they are of different sizes. Sludge sampling, which occurred in May and July of 2023, involved sampling sludge after the dewatering stage and sometimes before the belt press stage, using amber vials, transporting the sludge in cooler boxes that were full of ice, and then storing our sludge samples at 4 degrees Celsius in the laboratory within 7 days before extraction. On-site measurement included the measurement of pH and electrical conductivity parameters using a prepared slurry of sludge with ultra-pure water in the ratio of 1:5. In the laboratory, our lab samples were heated at 40 degrees Celsius in an oven, homogenized by grinding with a person mortar before characterization using our top-of-the-range, state-of-the-art instruments that we have here at the Florida campus. Phthalate extraction involved spiking 50 microliters of our mixed stock standard solution into 2 grams of our homogenized sludge, which was in 10 mil dichloromethane solution, and then sonicating 3 times at 45 degrees Celsius for 15 minutes, combining the extracts, filtering them, and then drying them in an evaporator to about 5 mils. That the results were then taken for a cleanup in a cleaning column, and then completely dried again in an evaporator and reconstituted with 1 mil dichloromethane hexane mixture before filtering again and analyzing the concentrations using a GCMS instrument. The human health risk assessment estimation was conducted using about 6 equations, as you'll be shown in the next coming 2 slides. The first 3 equations were used to calculate the hazard quotient of each of the phthalates in the wastewater treatment plants, and the hazard quotient, which was greater than 1, suggested potential adverse health effects. The non-carcinogenic risk assessment of all the priority phthalates were calculated using the hazard index, and again, a hazard index which was greater than 1 suggested potential adverse health effects. The carcinogenic risk assessment was only done for 2 phthalates, which are diethyl 2-hexyl phthalate and benzobutyl phthalate, because these are the carcinogens amongst the 4 that we were investigating. And their risk was calculated using equation 6, and the scale below was used to determine the extent of the risk. Any carcinogenic risk which was less than 1 times 0 times 10 to the power negative 6 was considered acceptable, and anything which was above 1.0 times 10 to the power negative 6, but less than or equal to 1 times 10 to the power negative 5 possible risk, and so forth and so on for probable risk and definite risks. The pH values of all our sludge samples from the 3 wastewater treatment plants were all slightly alkaline, in the range from 7.94 to 8.17. This was not far from the expected pH values of aerobically treated sludge of 7.8. And when we compared to the acceptable South African standards for agriculture, they fell within the acceptable range of 5.5 to 9.5, which does not enhance metal solubility. The electrical conductivity was also good. It was less than the limit, which is 200 millisiemens per meter, and it presented moderate salinity, which was good, which is acceptable within the South African standards. And in this regard, using these 2 parameters, it was concluded that the sludge was of good quality compost. Our SEM results, they indicated that sludge samples from the 3 wastewater treatment plants contained basically 2 mineral substances, which are organic matter, which presented itself in the form of irregular, strongly modulating morphological forms shown by 1 and 4. And then also there were calcium iron phosphate traces, which presented themselves in the form of oval clusters. Generally, wastewater treatment plant number 1 showed to have more of the mineral substances compared to the other wastewater treatment plants. However, from all the samples that we got, they presented to, the sludge samples showed that there were good soil amendments to be used in agriculture within the South African standards. The EDX spectra showed that carbon had the highest weight percentages in all the wastewater treatment plants. And this was expected because sludge has a high carbon content. And this was also good because carbon is food for soil microbes that bring nutrients for the plants. And then finally, on the characterization, the bolded values in all the respective wastewater treatment plants were above the South African Department of Water Affairs and Forestry Department total maximum threshold. However, they were below the maximum permissible level, and overall, all the sludge samples, they fell as pollutant class A, which is acceptable for agriculture within the South African standards. When it came to the occurrence of the priority phthalates in the sludge, all the 4 were detected in all the sludge samples from all the wastewater treatment plants. Their frequency of detection were all 100%, except in wastewater treatment number 3 for diethyl phthalate. The concentrations, they ranged from 643.90 to 1,981.28. Generally, wastewater treatment plant number 1 had the highest concentration of phthalates, and the lowest was in wastewater treatment plant number 3. The concentration distribution was in agreement with the octanol water partition coefficient, and the distribution of, however, the DAHP concentration was above the EU limit of 100 milligrams per kg. Overall, the concentration of the phthalates in the 3 wastewater treatment plants in July, which was winter, was higher than what we saw in autumn, which was May, and this was attributed to the load shedding that happened last year, which affected the operation of wastewater treatment plants because most of them, they do not have any generators, and also to the anthropogenic activities that are related to winter season. And of interest was the graph for wastewater treatment plant number 3, which showed an efficiency in the treatment system of that plant because there were little variations in the seasonal concentrations of the phthalates. When it came to the non-carcinogenic risks, the hazard quotients and hazard indexes of all the priority phthalates were less than 1, although they were higher in July. And because for the purpose of this presentation, we only used the results from the adults, from the male adults that we got, we then concluded that adults are unlikely to experience any potential effects from the above priority phthalates. And then finally, the carcinogenic risks for DHP, they ranged from acceptable risk to probable risk, while those for BBP, they ranged from acceptable to possible. In conclusion, the South African wastewater treatment plant produced sludge is generally suitable for agriculture, for utilization in agriculture, and all the 4 priority phthalates were detected in significant concentrations. Adults are unlikely to experience any possible potential effects from those investigated phthalates, and however, we have varied carcinogenic risks. And then future work could involve investigating the potential effects in children and investigating the priority phthalates concentrations in different commercially grown plants in soils that are amended with sewage sludge. These are some of the references that we used. I would like to acknowledge my supervisors, Prof. Sagati and Prof. Ngole, my mentors, Unisa, Institute for Nanotechnology and Water Sustainability, the Department of Chemistry, and the sampling team. Thank you for listening.
Thank you, Ms. Muzeza, for the very interesting and informative presentation. We now move on to the questions, colleagues. Questions, comments? Any questions from the floor?
Thank you.
Okay, thank you, Celia, for the good presentation. I just have a few questions. Maybe something I missed in the title, because I thought it touches on agricultural soils. I'm not sure if I missed that, because one of my questions will be based really on the statement in the title. But while you're still going there, don't you think with the concentrations you found, some being high in winter compared to the autumn, it's just maybe the dilution factor, because in winter we don't really get rains. It's not really related to the energy that, the low shading. Maybe when it rains, during the drying beds, some of these pollutants could have left the system. Did you think that could be one of the cases?
Thank you for your question. Okay, thank you for your question. Not really, because this sludge sampling was not only done once in these wastewater treatment plants. We did it several times in the 2 months that we talked about. And we noted that when there was load shedding, what would happen, it would basically be waste in and waste out. Because the wastewater, it trickles in using gravity. So it keeps on coming, and for that 2 hours when there's no electricity, the process continues, and all this, we get the byproducts and we get our effluent getting into the rivers. Unfortunately, that is what it is. That is what it was last year. So that was, in as much as what you're raising could be possible reasons, but this one was also a very important and main reason to take note of. Yes.
Yeah, there's a statement I just want to be sure before I ask in the title.
Okay.
I'm not sure it's not presented.
Did you say the question? Or?
Okay, so with the health risk in agricultural soils, do you think the pollutants availability, which can lead into the system of human beings, will not be a function of the soil mineralogy that is there? Because if the absorptions phenomenon are carrying, we might have those pollutants in the sludge, but never be available into the crops.
Sorry, come again, the question? The question?
No, I'm saying, don't you think
Yes.
the pollutants are there, right? But them passing into the food chain, won't you think maybe it will be a function of soil mineralogy? Because if we apply and the absorptions are there, depending on the clay mineralogy, we might never even think of the risks, because they might be adsorbed to the soils for a given time, and we should never be worried about them. Don't you think them being passed from the sludge to the food side, won't you think it might be limited by the soil mineralogy that exists in a given land? Thank you.
I'd like to agree with you to a certain extent. Of course, the types of soils, they should play an important role in as far as the absorption of these pollutants into the plants and eventually how they will then pass on into the food chain. However, it wasn't part of the scope of this presentation or of this work that we are doing. But what we need to appreciate is that priority phthalates are very trace compounds. They occur in minute concentrations, but they are so effective in those minute concentrations. We do have safe limits, though, that you can absorb into your system, and they will, as a result, not cause so much risks. That's why we had that grade of saying acceptable, possible, meaning they may or they may not likely cause any potential effects, probably meaning it's most likely, and then definite meaning it definitely will cause. And of course, I think you'd agree with me that the extent of the risks will also depend on your health as a person. But over and above, to answer your question, of course, the type of cell does affect the rate at which these pollutants will be absorbed through the sludge into the plants, and eventually how much we will get as top consumers in the food chain. I hope I answered your question. Okay.
Thank you. Thank you.
Thank you so much.
Thank you. A round of applause, colleagues. Thank you, Ms. Muzeza. Now we're calling upon our next speaker, Mr. Makwarela Tshireletso again. Hopefully everything will work smoothly now.
Wait, what? Switch again. Switch.
Can I please switch on to this?
Switch.
Thank you guys for your patience. I think I'm gonna just take over where I left. I would like to take it that it was a commercial break. Thanks. So as I was saying, the reason why I decided to pursue the study, it's because that ticks and the diseases that they cause on our livestock causes so much losses if we were to look at the figures. And so to look at their diversity and the distribution across the country, it's very significant in order to implement effective control strategies. So in order to get answers to our research questions or to achieve our objective, we decided to collect ticks in 6 different provinces, and those provinces displayed, or the regions in concern, or the study places, they displayed very different agroclimatic conditions. Ticks were collected in communal deep tanks, and then they were then transported to the Unisa, where we performed macroscopic observations to check their morphological features. We use the identified ticks then to extract DNA, and then which we amplified using the 16R DNA gene marker. Additionally, we took 4 tick species and then sequenced for complete DNA complete sequencing, which is our NGS. So the first objective, after collecting something, you want to identify what it is. And then the traditional method that is used, it's just the macroscopic one. So as detailed yesterday from Prof. Nyangiwe's talk, we use morphological features in order to classify them to their genera, genus, and species. Mostly we look at, I hope this is a pointer, I don't want to make other mistakes. So we normally use, let me just take this one, laser pen. So we normally look at their colors when we identify them, the presence of festoons at their margins, and then if they've got long mouth parts or short mouth parts. And then we also look at their legs. Some have got pale legs, while some have got bended legs. And then when we look at the ventral side as well, they've got different features, such as the adrenal plates, while some do not have adrenal plates. Some present some hairy-like structures, while some do not have. So after we have classified, we were using the key references from Walker 2003 and also Horak 2018. Where after classification, we found that from our collection, we had 14 tick species, of which it was Emblyoma herbarium, Haemophilus silacia, Hyaloma truncatum, Hyaloma marginatum, Axodes pilosus, Rhizophilus decolorata, Everti, Exothalamus, Glabroscutatus, Macroplas, Appendiculators, Cymas, Getridae, and Sanguineus. So the second objective was then to link the identified ticks from their different regions with the climatic conditions of those regions. So as there was a question yesterday from one of the judge that does the climate change have an impact on the ticks? Of course, as Prof. Nyangiwe explained, yes, it does. But when it comes to species to species, also there's optimal temperatures, optimal humidity, and optimal precipitation that is required for the tick survival. So we looked at the temperature, we looked at precipitation, and also we looked at humidity. What temperature does is that it influences the tick survival and development. When temperatures are very warm, it means that their productivity and metabolism can be increased. Therefore, we get more production of new ticks. That's why we find more ticks in summer and not in winter. So in winter, they can become dormant or even die due to low temperatures. So warmer temperatures favored the activities of Emblyoma herbarium in Limpopo, as they contributed 53.8%. And also 30% in KwaZulu-Natal, while the Hyaloma truncatum as well was favored by the warmer temperatures in KwaZulu-Natal. They contributed 50% of the collection population, as well as in Limpopo, they contributed or accounted for 34.6%. When it comes to precipitation plays a very important role in the habitat and host availability. As I've said, ticks are very fragile, so we need enough rainfall, or they need enough rainfall to ensure that the habitat is moist. So in moist areas, when they have like a hide in moist area or under leaflets or in humid places, it means they cannot go under the process of desiccation or drying out and eventually die. So region also with higher precipitation or rainfall, what they do is that they lush vegetation which shelter the ticks and also attract host for grazing. And Decoloratus was very favored by the prevalence of higher rainfall. In the Eastern Cape, their collection accounted for 73.7%, and also in KwaZulu-Natal, the total collection of this species was 26.3%, while Rhizophilus everti had a broader distribution in moderate rainfall regions such as in Limpopo, where it accounted for 38.0% and in Gauteng for 24.5% respectively. And when it comes to humidity, humidity plays a very huge role in tick physiology and behavior. As I've described that they need to keep or maintain their water balance, they also quest for host or for blood meal during humid conditions in order to avoid drying out. So there was a very high level of infestation of microplasts in the KwaZulu-Natal and also a herbarium in the KwaZulu-Natal due to the high humid level. And then we detected fewer ticks around the area of Free State, because it's very less humid, or the selected study sites were very less humid. So this indicates that ticks thrive less in drier conditions. Our third objective was then to confirm, because Prof. Nyangiwe mentioned yesterday, some ticks share overlapping characteristics. So we then had to use the 16R marker in order to confirm our morphological identification. From the tree displayed there, the colorful one, we found that every tick species clustered distinctly different from the rest. However, the species of Emblyoma herbarium and also Rhizophilus macroplus showed an internal sub-branching and genetic diversity based on their origin. This signifies that the geographic origin of ticks influences their genetic makeup. And we also confirmed this by the PCR, or principal component analysis, where we found that their Ixodes pilosus, because they were collected in one study area, they overlap or formed a very tiny cluster showing that they've got homogeneous genetic makeup, whereas the Emblyoma herbarium and then the Rhizophilus macroplus form a broader clusters that showed genetic variation. And then our fourth one, is my time still fine? Okay. Our fourth objective was then just to assemble draft genomes of 4 economic importance ticks, which was the Rhizophilus everti, Emblyoma herbarium, and R. appendiculators, also the Hyaloma truncatum. From using the Nova 6000, we were able to generate 120 gig of reads from the R. everti, and then for Emblyoma herbarium, we generated 107 gigabase data, and then for Rhizophilus appendiculators, we generated 117 gigabase data, and then for Hyaloma truncatum, we generated 117 gigabase. And then the estimated genome size for each species was 12.98 MB for Emblyoma herbarium. I think this one had an error because most of the DNA contained the host DNA, and then the reads for the actually Emblyoma herbarium was very small. And for Hyaloma truncatum, we were able to estimate the genome size to be 1.5 gigabase, and then for our appendiculators to be 1.96 gigabase. For our everti, it was 1.8 gigabase. And then the GC content throughout or across all the tick species, it was the same or almost the same. Thank you so much. And the duplication rates ranged from 9% to 11%. We then also predicted the proteins, where we found that R. everti had more proteins than the rest. Then the key findings are that indeed there is genetic diversity even within the same species, but it differs from region to region. And then Emblyoma, which causes the heart water disease, is the most predominant one, especially in Limpopo and KwaZulu-Natal. And then also, when it comes to morphology, it is very important to include molecular markers because some species do have overlapping characteristics. And then what I can then advise, what should then we do about this? I think we need to start developing acrosides that are tailored to different regions. We cannot just generalize. And then also we need to start teaching our farmers about the acroside use. Some of them are not knowledge about it, and then the dosage differs, and then the ticks end up adapting to different acrosides, and then therefore gaining resistance. And then in terms of biological, because there was a question yesterday as to can we use then biological controls? Yes, we can use biological controls such as the guinea fowls and the chicken. We have seen them in communal farmers unplugging those ticks. And then we can also do studies that involve the use of nematodes, because there are some worms in the soil that can release this bacteria that is very lethal or toxic to the ticks. We can also use some fungi. Some fungi produce spores that can actually enter into the exoskeleton of ticks and then grow inside and eventually kill the ticks. I think my presentation will end up here. So for this, we have done several publications. 2 have been accepted, 2 are under review, and then we have attended local and international conferences. Thank you.
Thank you, Mr. Makwarela, for the very interesting one. Oh, I see live animals here. I'm scared. Okay, colleagues, it's time for questions. I'm going to allow only 2 from the floor. Any questions for Mr. Makwarela? Comments?
Thank you very much, Mr. Makwarela, for your presentation. I just have some advice for you. I think your tick was disturbing me from watching all your slides. Unfortunately, my friend, it was a good idea. But if you only showed your tick in the introductory slide and stopped it for the rest of the slide, I think that will be a big favor for all of us, and if you want to present this somewhere else in future. Please.
Thank you so much, Prof.
Thank you, Prof. The last question, colleagues. There's one hand this side.
Yeah, thank you. I wanted to check with your genome, because you showed that you did genome assembly, of which you didn't report anything relating to the genome size which you got. You just mentioned the estimated genome assembles for those species. So I will want to find out from you, in terms of the size, how much did you get? And again, you also reported something related to the protein genes. So how did you observe, how did you acquire those protein genes? Thank you.
Okay, so the annotation protocol or softwares that we've used for protein prediction, it was Busco. Doctor, are you saying something? Oh, okay. We use Busco for completeness of the genome assembly, and then also for protein prediction. However, we didn't do further annotations, of which we are busy with now for publications. Thank you.
No, can you use them? Give him.
Yeah, please. Sorry, this is to the chat. It's not to you, Makwarela. Oh, okay. I just wanted to find out, was the time, maybe Ray should also assist, was the time allocation for the PhD candidate altered? Because I thought they were given 15 minutes for presentation and 5 minutes for questions.
Yes.
But
no, we're sticking to time even now.
Yeah, but
it seemed like we gave
stopped him very fast.
Is it? Hard. But my time was okay. No, I could really the mistake. Okay, thanks, colleagues. Thanks, Mr. Makwarela. A round of applause, please. Our next speaker is going to be Mr. Lazarus Katlarumrani. time yourself. It's 20 past 1. I can they say we're not giving you time, so it's 20 past 1.
Is this your presentation?
Good afternoon, everyone. Before I begin, I would like to start by telling a story. A couple of years ago, I got a call in the late hours, around 10:30, and the call was about kids having similar symptoms who attend the same school. And when we got there, we actually decided to take samples to the lab, and we analyzed them. Unfortunately, we didn't know what was the problem, but based on the symptoms that the kids were presenting, we had ideas, but still we didn't know what was happening. In the morning, 10 kids were hospitalized, each presenting profuse diarrhea. The motive for that story is that our monitoring systems in terms of water quality in the municipalities are not really working for us. And I want to emphasize that water quality plays an important role in balancing ecosystems, aquatic ecosystems, and also making sure that there is safe drinking water for our communities. However, there are issues of water pollution from industrial discharges, agricultural runoff, and urban developments that poses a threat or challenges to the current standards that we have right now, such as the Science 241 or the RQOs in terms of water resources. As such, accurate and timely prediction of water quality is essential for effective management and for development of mitigation strategies that are timeless. Currently, the traditional methods for water quality monitoring often rely on periodic sampling and laboratory analysis, which are time-consuming, they are financially straining, and do not support real-time decision-making. As such, there is a growing need for more advanced and efficient approaches to predict water quality. Recently, machine learning algorithms such as ANNs have demonstrated success in various predictive tasks in different domains. ANN in particular have shown a great promise in handling complex data that has nonlinear relationships and data sets, making them an ideal choice for water quality prediction. As such, the objective of the study was to develop a water quality prediction tool that is based on an ANN machine learning algorithm. This tool aims to offer robust and reliable solution to predicting water quality indicators to enhance the ability to monitor and respond in time to potential water issues. So the development of such a tool holds significant promise in that it will improve water quality management practices in municipalities, in government, wherever. And ultimately, this will lead to protection of the human health and the environment. So to develop this tool, we had to source historical data from different sources. We used the IUCMA, Inkomati Usutu Catchment Management Agency, which is a CMA that is based in the Mpumalanga region and that manages all catchments in the Mpumalanga region, and they have data that they produce each and every month. And then we also use data from the South African Weather Services So the parameters that we chose, let me just get my point down. So the parameters that we chose, these ones, there were 13 in total, and they were based on the risks that are occurring in that area, in that region. And also according to the current RQOs in the water resource. So the second stage involved designing the actual ANN model from the data set that we could gather. So we split the data into 80-20, that is 80 for training and 20% for evaluation, that is testing and validation. And then from there, we set on to determine the parameters of the training in terms of how many neurons are we going to include in there, and how many hidden neurons are we going to include, how many activation functions are we going to use, or which ones are we going to use. But this was actually a trial and error method, because training, even if you predetermine the parameters, it's really difficult for you to know whether it's gonna work or not. So it's better to always use trial and error. So for evaluation, we used 4 regression error models called mean square error, mean squared logarithmic error, mean absolute error, and mean absolute percentage error. So for an error to be acceptable, or for the model to be acceptable after training, the error has to be less than 0.5. So when we decided on the architecture of the model, we used a fully connected multi-layer network that can produce either one output or many outputs. However, if you have more outputs, it means that you're going to require more data for you to train. So when we started with just one output so that we have enough data to train the model. This is the prototype of the model, actually the tool, not the model. So for you to get here, you first need to have your raw data, and then partition your data in terms of what is it that you want to do with it. How much data will you use for training, and how much data will you use for validation and testing. And from there, you'll set your parameters in terms of how many input neurons are you going to use, and how many hidden layers are you going to use, and then you'll train your data. So training will normally take around, on average, 15 minutes for data that is normal. But for advanced models, you'll have 30 minutes minimum to train your model. So these are the training results from the physical parameters that we had, and we only had a failure we were only unable to produce a reliable model for one in terms of the physical parameters. So the closer the graphs, the more reliable the model. So the pink one is actually a validation curve, while the blue one is the actual training. So validation is mainly an act of proving accuracy of something. So if the validation curve is closer to the training curve, it means that the model is actually acceptable. I'm just going to rush through. So the one that we actually had problems with, which even today I can't explain, is the electrical conductivity, because most of the data that we got for pH and turbidity and temperature were almost the same, almost in terms of quantity were the same, in terms of electrical conductivity. But the actual model didn't work out. As for all the chemical parameters, we all got reliable models. But the interesting part happened when we tried to test the biological parameters. And all of them, from total coliforms, E. coli, fecal streptococcus and bifidobacteria, all of them fail to produce reliable models. But these are the best ones we could get. Although they are unreliable, we still cannot use them for testing or to use in real-life situations. So we actually attributed this to a lack of data and maybe training parameters. From the data that we got from the training, we actually are assuming that there are two main aspects that are contributing to the results that we got. First is the data splitting. So if we used actually 80-20 in this study, and we realized that the more we increase the training data from 30% to 80%, the more accurate the models were, and the more reliable the models were. However, other people found that if you increase more to 80%, from 80% to 90%, you actually have a different trend in terms of your reliability of the model. The second one was the activation functions. Activation functions actually starts the actual training. So without the activation functions, you will not have training. So the choice of the activation function that you're going to use has to be aligned with the type of data that you have. So in this case, we used two activation functions. One is an actual traditional one, and one is the actual new kid on the block, the one that everybody is using. However, it was interesting that in our results, most of the reliable models that we got were actually generated using the traditional sigmoid activation function. In conclusion, we acknowledge that all forecasting models are data-driven, and with increasing accuracy, you require more data inputs. And maybe that is the limitation of this study, to say maybe there needed to be more data that we needed to get, especially for the biological parameters. However, we are proud that we could produce 62% of reliable models from all the parameters. And as such, we urge that further work on this tool or on a similar study emphasize on acquiring more data that is related to biological parameters. And on that note, I would like to thank my supervisors and the institution. Thank you.
Thank you so much, Mr. Morani, for a good presentation. Morani, right? Thank you so much. Colleagues, we are running out of time and will only allow one question. We can engage further with our students during the lunch break. Just one question? So I'm saving you. There's a hand here.
Thank you, Katlego. So mine is just very straightforward, because I'm just in the discussion, the EC model is not doing well. So I just wanted to know, in the parameterization of that model, did he have assumptions? Because I could think maybe the EC values will depend on the hardness of the soil, of the water. So was this specific in the parameterization? Did the model have specific assumptions just to cater for the hardness that you might get when you're measuring your water? Thank you.
Thank you for the question. We didn't have any assumptions. Remember, you normally get hardness, actually great hardness in drinking water. That is maybe from groundwater, actually. So this was surface water, so you'll not really have much hardness from surface water. So we didn't really have anything to do with that. Yeah.
Thank you so much. Can we give Mr. Muhani a round of applause? Our next speaker is Ms. Masalauno Mukadi. The floor is yours.
Dima siari. Zinalanga ndifi musara unome simkadi. As introduction has been made on ticks, I'm just a sardine swimming with the sharks. I'm a master student. That was the typo. Then my study here is going to focus on molecular prevalence of bovine babesiosis in cattle population of Mutare local municipality in the Bembe district, that's in Limpopo province. The sharks have spoken already since yesterday. Under the supervision of Dr. Belona Sakong and the co-supervisor, Dr. Sikumbuzo Mbizeni. What is bovine babesiosis? In the Limpopo province, our grandmothers called it chihebe, while it is suck blood already, ndigupa. Then in layman's English, it is red water. This disease was first found in the year 1888 by a man known as Victor Babes, hence he named it after himself. It is also known as red water that affects cackles. It is caused by one or more intra-erythrocytic protozoa of the genus Babesia. Babesia has got vast species that are found in it, which are Babesia bovis, Babesia bigemina, Babesia divergens, and many other species. But in this study, we shall focus only on the two, which are Babesia bovis and Babesia bigemina. Why? Because it has been found to be prevalent in South Africa through the past years. That's according to Kivaria in the year 2006. And again, it was put again into FRC by Nyangiwe and the others in the year 2017. The direct effect on production on bovine babesiosis is visible because the tick mounts to the cackle, and the cackle becomes restless. And the farmer, the communal cackle farmers, tend to see that with naked eyes. While economically, it becomes strenuous to communal cackle farmers because they have to dig a hole in their pocket so that they can send their cackles to deeps and also to acaricides sites. Then the indirect losses, they affect me and you through the red meat industry. That's why our chisanyamas today are very expensive. Our milk also. Some families are eating cornflakes without milk because those are the indirect losses that we see. And then you can ask yourself, what is the problem? The problem that has affected the study area is mortality and morbidity. Because in sustainable stocks and also in crossbreed cackles, this disease has become prevalent. And then the transmission and the occurrence of these tick-borne diseases is only vulnerable to the tick infestation due to the lack of proper tick control measures. But it also increases the occurrence and the transmissions of the tick-borne, as is stated by Murife. And then the clinical disease indicative of this disease are continuously reported by the communal cattle farmers, although there are still no clear records that are stating the prevalence and their tick vectors in the area. Then the economic significance of these tick-borne diseases, there's still a need to get the true prevalence and the distribution. It's still just a talk of town, but there's still nothing that has been done. What is the aim of this study? It is to gain better understanding about the prevalence and the risk associated with Babesia bovis and Babesia bigemina species in the Kambule heads of Motale local municipality in the Bembe district. For us to achieve the aims, the following objectives were set, which the principal objective is to determine the molecular prevalence of bovine Babesia species, which are Babesia bovis and Babesia bigemina in Kambule heads of Motale local municipality. The second objective is to determine the prevalence of tick species, the Rhizophilus decoloratus and the Rhizophilus microplus, that transmit the disease in the study area. And the last objective is to investigate the risk factors that are associated with the occurrence of bovine babesiosis in Kambule heads of Motale local municipality. I would like you to stop asking yourself many questions. I have questions set for you that you might focus on them. What is the prevalence of bovine babesiosis in the study area? It has been found to be prevalent from the previous studies, as the big sharks have spoken since yesterday, in the study area, Motale local municipality. What is the prevalence of ticks in the study area? The study area has been found to be more prevalent of different species of ticks. What are the risk factors associated with the occurrence of bovine babesiosis in Motale local municipality? The risk factors have been affecting communal cattle farmers rapidly. This is the study area. It's Motale local municipality in bold circled. This area received approximately 600 millimeter rainfall per annum, with cattle head population in the state veterinary offices recorded to approximately 37,000, and it carries about 123 villages and with 50 deep tanks. But for this study, we shall randomly sample only 20 deep tanks because some are no longer in good state use. They are no longer well taken care of. And then about five of them are in the proximity of the game reserve. As we know that ticks mount in all circles, so those that are in the proximity of the game reserve, we hope to get higher amount of tick species to check the Rhizophilus decoloratus and the Rhizophilus microplus. The sampling methodology in this study has been taken out from the calculation by Threshold in the year 2018 for the large sample size. There is also a sample size for small sample size, as we know that mortality and natality happens every minute in animals. So therefore, the sample size that is going to be calculated according to the formula as stated there, whereby the expected prevalence is at 50%, nothing less, but something more than 50%. And then the desired level of precision at 5%, whereby the Z statistic for this level of confidence at 95%. The cackles will be restrained in the crash pen before handling and taking of blood samples and the ticks. And all animals will be randomly sampled to the participating deep tanks following the predictor variables, which is their age groups, the sex, the breed type, body condition scores, tick infestations, geographical area, and also again, biosecurity control measures shall be taken into consideration and be administered in all animals that will be participating to ease the spread of the tick-borne diseases, because we hope to get the prevalence. Therefore, when all the samples have been taken, these other laboratory analyses that are going to be performed, whereby the DNA extraction will be used using the whole blood sample of the Zymoquit DNA mini prep kit, where the molecular assay that is going to be performed is the qualitative polymerase chain reaction assay for the Babesia bovis parasite detection, whereby the DNA will be amplified into primers as described by Bria Ruhanga in the year 2023. And then for the Babesia bigemina parasite detection, the DNA will be amplified following the primers as described by Stolz in the year 2020. And then the following table is for the primers and probes, whereby it's going to be used according to the Techman polymerase chain reaction of the 18S RNA gene, whereby no primers and probes shall be designed. All the primers and probes sequencing shall be used, those that are existing with a gel electrophoresis, following all the sequences that has been done for Babesia bovis by Buranga and for Babesia bigemina by Stolz. Then the emancipated results for this study, we expect to find the prevalence of Babesia bovis through the Pearson chi-square tells will be used to evaluate the association of the molecular prevalence, the qualitative polymerase chain reaction positivity of bovine babesiosis with the explanatory variables that have been followed in the study area. And the second one, we hope to get the prevalence of the tick species that transmit the disease with the Rhacephalus decoloratus and the Rhacephalus microblast in the study area through the Pearson chi-square again test that will be used to evaluate the association of the prevalence. We will use the one-way ANOVA to compare the mean tick density in all animals, and the ticks will be taken from only the one side of the animal body, whereby the prevalence of the risk-associated factors is also the result that we're hoping to get, whereby one-way analysis of variance, the ANOVA, will be used to compare mean density. And all the calculations, in all the calculations, the confidence interval and the significance interval will be set at 95% and at 5% respectively. We hope that the study will reveal the significance in undertaking the epidemiology of animal health and the accountability of ticks and the tick-borne diseases by the communal cattle farmers in the study area, and thus will also assist to provide the relevant information from cattle farmers and the veterinary authorities to develop control measures and ask researchers to come up with new methods for acaricides and to ease tick-borne disease spread in the study area. And the biosecurity precautive measures will be directed to all the sampled animals, whereby the results and findings will all be addressed back to the farmers through their community engagement, Farmers' Day, Communication, Information Day, and the Farmer Support Outreach, with the liaison with the State Veterinary Office and the Agricultural Extension Officer in the study area. I'd like to thank my supervisors for their endless advices and support in this project as it's still proceeding, and my sincere gratitude to the University of South Africa for the financial assistance at hand. Then the reference for all in the study I shown, Ndoribua.
Thank you so much, Ms. Mukadi. Colleagues, we will allow 5 minutes for questions.
Thank you. No, mine is just a simple one because I just wanted to find from that one-way ANOVA, what is that independent variable that informed the one-way ANOVA? Thank you.
Thank you. It is the mean tick density.
Yeah, maybe for just an advice, we no longer have Thulamela municipality, so make sure that you change that. There's no Thulamela local, Mutare local municipality now.
Okay, thank you. Beforehand, prior, the study was going to be conducted in Thulamela local municipality. Then because of FMD, foot and mouth diseases, then it will disturb our results. So pardon me for not stating that initially. Then Mutare local municipality is now jointed 50% with Thulamela and 50% with Musina.
Yes.
So all the villages that was collected was from the previous Mutare local municipality. So the study is going to shift to Musina municipality, not the whole 100%. I'm not sure if I'm clear. Okay, thank you.
Thanks, Chair. I'm happy with your laboratory analysis for your first objective or aim. But for objective 2, which is to actually determine the prevalence of Rhebkephilus urticulatus and Rhebkephilus microplus, you didn't tell us how are you going to analyze that? How are you going to differentiate between the two species, or the methodology that you are going to use to collect those species?
Thank you. For objective 2, which is to determine the prevalence of the Rhaciophilus decoloratus and the Rhaciophilus microplus, those species that transmit the tick-borne disease in the study area, we're going to use the tick identification method as you have explained from your study. I'm going to proceed. I'm taking my steps towards. I'm starting swimming with the sharks.
Good afternoon, and thank you, Chair, for the opportunity to ask the question. And congratulate to the student. Mine is just a simple one. I just wanted to find out, since you were collecting the ticks from those municipalities, so I want to know the advice to the farmers. How would you advise the farmer that they were affecting with those ticks in those municipalities? Thank you.
Thank you. Based on the previous records, farmers could see symptoms that are happening on their animals, but they couldn't voice out because they were unfamiliar. Hence, that's why, on the conclusion, farmers were going to be educated through their farmer support day in liaison with the State Veterinary Office and the Agricultural Extension Offices. So for farmers to be knowledgeable, as the aim says, is to gain better understanding and knowledge about the prevalence of the ticks. We all know about ticks, but we don't know the danger that ticks causes. So we're going to give information through the State Veterinary Office.
So I just wanted to find, while you are busy collecting those ticks from the animals, so you were with the farmers, I just wanted to hear from you, what did you advise them? Because you were collecting with the owner of the animals. So how did you advise them in terms of, because those animals, remember, they were getting injury. Immediately when you remove the ticks, they get injuries. On some of okay, thank you.
Farmers were advised to follow animal health protection programs as stipulated by the State Veterinary Office. Animals were getting injured, yes. Biosecurity measures were administered immediately after the tick collections. So we never diverted from animal health protection programs. We stick to them, and also inform farmers about all the risks associated to report them to the State Veterinary Office.
Thank you so much, Ms. Mukadi. A huge round of applause for Ms. Mukadi. Our next speaker is Mr. Malindisa Sibusiso. The floor is yours.
Good afternoon, colleagues. I understand I'm the one between you and your lunch, so I'll try and do my best to finish quickly. Can I get a pointer? And I promise I won't have any genus names in my presentation, so no vetsi. Great. Perfect, thanks. And then changing slide, this one. Perfect, thank you. Cool. So good afternoon once more. I'm Symposium Marindisa, a PhD student under Prof. Ndwasa, and today I'm going to talk to you about one of the 3 drugs that I was testing for my PhD, and that drug is ezetimibe. So the title of my talk, therefore, is the in vitro analysis of ezetimibe-induced cytotoxicity in melanoma and lung cancer cells. So what is this ezetimibe? So ezetimibe really is a drug that is used for lipid lowering. So it is used basically to reduce the amount of cholesterol in the blood. It binds to this particular, it binds to this, it's not happening. Oh, there, okay. So it binds to this particular receptor, which is responsible for transporting lipids between cells. Now, this particular drug is used to prevent cardiovascular diseases in people who suffer from high cholesterol, which is hypercholesterolemia. Now, this work is based on the background that in silico work that was done by Prof. Ndwasa, which they found that this particular drug binds a particular protein called MDM2. So MDM2 is a well-known negative regulator of P53. So some of you may know already that P53 is the guardian of the genome, which means whenever there's something going wrong in the cells, P53 is usually activated to cause apoptosis or cell death. Now, P53 is tightly regulated by this gene called MDM2. So you can see here, P53 is tightly bound to MDM2, and this is because to ensure that cells don't die for no reason, okay? So it's to basically limit the expression of P53 in normal cells. However, we have seen that this particular pathway is very dysregulated in many cancers. About 85% of known cancers have a dysregulation in the P53 pathway. So therefore, making it very important for us to target. Now, ezetimibe binds MDM2 at the P53 binding domain. So now we predict that if MDM2 can bind there, it can disrupt this interaction, allowing P53 to be released and then cause apoptosis in cancer cells. Now, why is this important? Well, because cancer is a global issue, okay? Many people are dying of cancer globally, and therefore we need good diagnostic and treatment options for everyone. So if you look in this graph, you can see that there's high incidences of cancer in most countries, and the side shows you the mortality. So if you look closer in countries like Australia and New Zealand, there's very high incidence of cancer, but look at the mortality. The mortality is much lower than the Asia continents as well as Southern Africa, which means then there is a disparity between these regions, between low and high income countries. So therefore, it is therefore important for us to find other drugs that are accessible to low and middle income countries. And this strategy is therefore drug repurposing. So therefore now we use drugs that are already approved for other diseases, ezetimibe, to treat cancer. And this process is a very nice process because usually drug development takes about 15 to 20 years from basic research all the way to the drug being approved. It takes about 20 years and it costs a lot of money. So if we repurpose drug, we reduce this whole cycle of new drug development by a number of years, but also we reduce it because the drugs are already known, their safety profile is already known, therefore many of these tests are not required. And also this drug repurposing also helps with costs because it reduces the cost of producing a drug. So I can be a billionaire much quicker. So the aims of this project really was to validate the anti-cancer activity of ezetimibe, and we did this firstly by doing MTT assays to ensure, to test the cytotoxicity of ezetimibe and determine the inhibitory concentration. We then did flow cytometry, particularly apoptosis determination and cell cycle, to understand the mechanism by which ezetimibe induces death. And then we also looked at the number of proteins that are involved in proliferation, how the expression is affected by ezetimibe, and then finally looking at some of the drug targets. So I will not talk about the last one because that one is more in vivo, and because of time, I'll just stick to the form. So briefly, MTT assay involves the detection of this particular enzyme called, this particular molecule called tetrazolium. So usually when cells are live, they can convert this tetrazolium into a formazan dye, and this formazan dye is usually purple. So what we do then, we treated the cells with the various concentration of the drug, and these are the cell lines that were used for this study. Now it's important that we also understand the status of P53 in these cell lines, because if P53 is mutated, then reactivating it via MDM2 will not help us. So then from the MTT assay, you can see that ezetimibe was cytotoxic to lung cancer cells, which are A549, A375, which is a melanoma, and it was not toxic to the normal cell line, which is HEC. So if you look at these two cell lines, PANC1, which is a pancreatic cancer, and colorectal cancer, both of these have mutant P53 and were both unresponsive to ezetimibe, which then took us to our next experiment, which only limited us to only these two cell lines that were positive for ezetimibe. So as you can see from this image, that treatment with ezetimibe firstly reduces the number of cells, but if you look at these dark spots, those are condensations which also indicate that there's apoptosis happening. Now, after that, then we wanted to know how are the cells dying by flow cytometry. So this briefly involves staining of the cells with annexin 5, FIT-C, and propydium iodide. These two allow us to detect cells that are undergoing early and late apoptosis. Now, from the results, we can see that treatment of the lung cancer cells induced apoptosis in treated cells, and you can see here an increase in apoptosis compared to untreated cells. So if you look at both also the early apoptosis, you can see that at least over 60% of the cells were undergoing apoptosis, whereas the untreated cells remained, at least 60% of them were still alive after 48 hours. We did the same as well for the melanoma, and again we saw the very same phenomenon where in this case, most of the cells were in the early apoptosis. Now, from there, we also wanted to find out how does ezetimibe affect the cell cycle. And again, we used a similar method of flow cytometry, but the only difference is that now we only stained with PI, which only stains DNA. And because of that, we can now therefore use that phenomenon of PI staining and DNA to detect in which stage of the cell cycle the cells are following treatment. And from the results, we can see already that there is a shift from the G2M phase to more of a G0, G1 phase. The same we also saw in the A509 cells. Now, what does that mean? So therefore, we wanted to see which proteins are participating in this process. So we performed an automated Western blot, which briefly involved treating the cells with the IC50 concentrations, and then extracting the protein, measuring the protein concentration, and then running it on this very nice automated Western blot machine, which reduces your Western blot from 4 days to about 3 hours. And the result from this work usually gives you a blot and also an electrophotogram showing where your cross-reactivity with your antibodies was. Now, we then measured the the protein concentration using the qubit, and we found that the protein concentration was very similar, and we also used the loading control to make sure that we are detecting the right concentrations. The first gene that we were interested in was MDM2 because our drug binds MDM2, but after several attempts, we could not detect it. So this still requires a lot of optimization. So then we moved on to the other genes that play a role in the P53 pathway, and one of those was P53 itself. So then we looked at P53, particularly the phosphorylated one, and we found that P53 is expressed in untreated cells compared to treated cells, which was very interesting because we expect our drug to increase P53. Now, the downstream gene that is activated by P53 is P21. We also did the same, and it had very similar results of downregulation rather than upregulation. So then, luckily enough, the kit that we used contained an EC1 antibody. So we were able to also detect EC1. EC1 is a kinase protein that plays a role in the MAP kinase pathway. The MAP kinase pathway is involved in proliferation. So high expression of EC1 means the cells are dividing. So after treatment, we've seen also again that EC1 seems to be downregulated in ezetimibe treated cells. So again, now we don't know what all of this means because we're expecting to see P53 and P21 upregulated. So then what we did is we performed DATS, which is a drug affinity response targeting stability assay, which briefly involves taking a cell lysate and then subjecting it to your drug. In our case, it was ezetimibe, and then having a control which does not have a drug. Now, you subject this mixture to protease degradation, and in theory, proteases should not degrade proteins that are bound by any ligands. And from there, then you run a polyacrylamide gel, and there you can detect which of the proteins bands are protected, and then we ran a mass spec to determine which proteins are those. So from our experiment, you can see that from the vehicle one, there's many bands that were digested, but there were these bands that you can't find here, number 1 and 2, and we chose these ones particularly because we thought, because MDM2 is around 50 kilodaltons, so we expected to see it there. But then upon analysis, we found that none of these targets are MDM2. However, what is interesting is that some of these targets play a role in cell proliferation, particularly the DNA homolog, which is associated with regulating BEX. So BEX is also one of the proteins that when cells undergoing apoptosis, it's upregulated. Another one is septin-7, which basically allows for the S-phase entry. You will see later on why we did not get to the S-phase entry. We also looked at alpha amylase, we look at many other proteins that we found that that ezetimibe potentially binds to and are also involved in the proliferation of cells. So, basically, we found that ezetimibe does induce cell death in lung and melanoma cells in a concentration-dependent manner whilst protecting normal cells. This, therefore, underscores the safety profile of ezetimibe. Interestingly, ezetimibe was not toxic to our p53 mutant cell lines, and it's not surprising because usually p53 mutant cancers are resistant to most types of treatments. What we also saw is that ezetimibe promotes apoptosis and also induces a cell cycle arrest at G0 and G1 phase. Now, at G0 and G1 phase, we expect to see a high expression of P21 because P21 is one of the genes that is upregulated at the G1 checkpoint. However, we did not see any P21. We did not see any phosphorylated P53. However, EC1 was downregulated. Now, what does that mean? It means that possibly ezetimibe is acting or inducing apoptosis in a P21 and P53 independent pathway. The fact that EC1 has been downregulated, it tells us something. EC1 plays a huge role in the MAP kinase pathway, and it is highly expressed in many cancers. So, it is also known to promote the activity and expression of cyclin D and E. So these cyclins are responsible from the transition from the G1 to the S phase. So now, it makes sense then now when we look back to our cell cycle results that we saw most of our cells in the G1S phase and clearly EC1 is downregulated, and when EC1 is downregulated, most of the cells will not proceed in that direction. The fact that these proteins have also been identified as potential targets for ezetimibe further supports this notion that ezetimibe induces a G1 halting of the cells, particularly if you remember from the table septin-7. So septin-7 promotes the transition from G1 to S phase. So if septin-7 is inhibited, the cells will not move on to the S phase or continue with the cell cycle. Casein kinase 2 also is a protein that acts as an oncogene when it's highly expressed. Alpha enolase also works in various pathways involving expression of various genes. So downregulation of alpha enolase can also impact the transition of cell proliferation. So, in conclusion, we have seen that ezetimibe is a potential anti-cancer molecule because it did show anti-cancer properties in these cell lines. So therefore, it has the potential to be repurposed for treating cancer. Currently, we are testing the drug in combination with other drugs for cancer treatment, and we have the first preclinical data, but at the moment I'm not at liberty to share it. But basically, ezetimibe can possibly be used for cancer treatment. I'd like to thank my funders and Tia also, who funds Prof. Ndwasa. And yeah, this is the team, and this was one of our cake days involving something else as well, but who am I to say? Thank you for listening, and questions?
Lisa, for the very interesting presentation. Let's take questions from the floor. Any question? Sorry. Not one.
Thank you, Sbusiso, for quite an interesting talk. I was just wondering, when you were showing the incidence of the cancer, is it the overall cancers or is it specific cancer incident? Yeah, because the thing why I'm asking, so some of the cancers, we already know that they are associated with factors such as dietary intake and, for example, colorectal cancer, in where maybe it's a good point to mention that we have seen that the traditional food, the traditional African food, it's actually protective of colorectal cancer. So I'm just wondering if we can assume that all of them, it's a similar pattern, that it's happening because of the lifestyle, why we see many incidences in the first world.
So yeah, that graph really was showing the overall cancer rates in the different regions. But the cancers that are incidents are different in those regions. For example, in Europe, lung cancer is one of the common cancers. In Asia, because a lot of people are smokers, so therefore the high rates of lung cancer will get there. But in other areas, like here in Africa, you'd see like stomach cancers, prostate cancers, which are the leading cancers, because there's not a lot of smokers in this area, you see what I mean? So these different cancers are based on regions and their lifestyle, as you've said.
Another question? Sorry? Yes.
Yeah, be brave, be brave. Have you thought of looking at things like caspase assays to prove that what you're looking at is apoptosis?
Yeah.
So they'll come next, but...
So we did caspase 9 as part of our Western blot, but then because something that we got recently, we're still optimizing it. I've done it 2 times already, but I couldn't detect it anyway. So it's more of an optimization issue that we're dealing with. Yeah.
Let's take another question, colleague.
It's lunch.
Yeah. The last one for Sbusiso. In the absence of any question and comment, colleagues, there's an announcement from our organizers. Actually, this is how things are going to be followed outside for our lunch. We're going to have our speakers and our adjudicators going first, and then we as staff will follow with the tickets. So if you don't have a ticket, you should wait for those ones with the ticket. And then if, fortunately, there's food left, then you'll get an opportunity to grasp one. Thank you, colleagues. We'll reconvene at 3 o'clock. Thank you.
Is it?
Can you guys hear me? Okay.
Good afternoon, colleagues. Can we please take our seats? Okay, welcome to the last session of the presentations. They always say we save the best for the last. So, colleagues, fasten your seatbelt. We are going to move very fast because of time. So I would like to welcome, before I welcome the speaker, I would like to introduce my colleague here, Dr. Van Onselen. And I'm Lidi Lemanga. I would like to welcome the speaker, Neo
Dr. Neo Mokhadi, on the topic recent advances and developments in bacteria endophytes, identification and application. It's a review.
Good day, ladies and gentlemen. My name is Neo Mamecha, a postdoc fellow in the Life and Consumer Sciences, under the mentorship of Dr. Tracy Masebe. Today, I am going to give a brief talk on a review we're working on bacterial endophytes, titled Recent Advances and Developments in their Identification and Characterization. Bacterial endophytes in the past 10 years have gained attention because of their important roles in different fields, be it your host, environment, and the lives of humans. These bacteria are defined as bacteria that live within plant tissues and cause no apparent harm to their host plants. They are present in every plant part, your soil, your seeds, your roots, your stems, and the likes. Bacterial endophytes colonize diverse plant species, forming a symbiotic relationship and benefit the host plant by enhancing nutrient uptake, stress tolerance, and disease and pest resistance. The three are just examples of endophytic bacteria, which are Picoderia, Phytophthirans, Pseudomonas putida, Acricoccus terreus. The survey methodology followed the PRISMA guidelines of sourcing publication articles and dissertations using keywords, bacterial endophytes, diversity, application, isolation, and their identification and characterization. Ladies and gentlemen, the study aimed at bringing to light the recent advances and developments in the identification and characterizations of our bacterial endophytes, and further highlight their challenges and limitations. Yes, Dr. Malobani, we are saying bacterial endophytes can be used as biocontrol agents where we will not need the use of chemical pesticides, and this will enhance the growth of our plants and make them resilient. And then in pharmaceuticals, we are saying bacterial endophytes can be used as metabolites that have anti-tumor and antimicrobial properties, because some of the microbial and some of the bacterial endophytes excrete metabolites the same as those of their plants, and this in conjunction can be used in drug development. This is just a quick picture of. I cannot point. Okay, on your right is a jar that has endophytes, and on my left is a jar that has endophytes. And on my right is a jar that does not have endophytes. The plant in our left, how do I do it? Okay, the plants on our left show that they have, with endophytes of course, show that the plants are healthier and resilient to a couple of stresses, and our right jar shows no endophytes, and this, you can see they are struggling. So ladies and gentlemen, endophytes are important in the sense that if a plant does not have endophytes, it's as good as dead, because there is no mechanism that will help the plant to fight stresses. This is also just an overview of the study. You can see on your left on my left is a plant with endophytes, and on my right is the one with no endophytes, and the one on my left shows that it can tackle the world because it's resilient and healthier. Okay, identification of bacterial endophytes starts by isolation, where they Okay. Identification will start with isolation of our plant tissue, where we need to sterilize the surface, and then that will need surface sterilizers, such as our sterilizing agents, our ethanol, methanol, and the likes, and also macerated in excessive water to remove excess sterilizers. Fragment the plant tissue.
Use this one to point, you use the computer to continue the slide. This one is just for pointing.
Okay, thank you. Fragment the plant tissue and then inoculate it, of course, incubate it under favorable conditions. Of course, we are going to get mixed populations of our endophytes. Then that's where we'll do serial dilutions to get pure isolates. Identification of endophytes is done in two techniques: your culture dependent and culture independent. Culture dependent only focuses on the culture and morphological identification, which is followed by biochemical activities to check their functional activities. Culture independent techniques, we have to now acknowledge that they've brought enough information or enough resources for us to characterize our microbial populations on a molecular level. This includes the 16S, which confirms the type of species, our PCR, which quantifies, and our omics, which gives an overview of the functionality of our metabolites. Quickly, this is just an overview of what I was saying about identification and characterization of our endophytes. First, you collect your sample. You have to sterilize it. For sterility, to validate sterility, we need to inoculate. After sterilization, we inoculate, culture, and if there's no growth, it means kudos, then there is no unwanted material. Then we can go straight into our characterization, which includes culture dependent and culture independent. I said with the culture dependent, it's our culture straight into the results and biochemical analysis. And then our culture independent involve molecular studies and visualization. Take-home message today is that studying endophytes requires combination of disciplines, which are your microbiology, botany, ecology, and genomics, just to name a few. And this evokes current knowledge we know of research for the future's perspective. And we acknowledge that it is going to be a challenge, and these limitations can highlight the need for continued methodological development, innovative approaches, interdisciplinary collaborations, of course with the throw of AI tools to overcome the challenges. With that being said, thank you for your time.
Thank you. Any questions? I would allow 2 questions. Going once.
Thank you very much. Is this working? Okay. The first slide you showed us, you said with endophytes and without endophytes. Am I correct? And you were defining endophytes as being something inside the plant. How did you know that the one set had no endophytes? How did you remove, how was the endophytes removed from that plant? Because I saw a 2017 reference there. Are they talking about endophytes inside the plant, or they were supplied those things?
Endophytes, Prof, are everywhere around the environment. They are inside the plant, they are outside, they are in the soil. So in some studies, it shows that most of them are more concentrated in the roots of the plants, which are bacterial endophytes, are more.
The word endo means inside.
Yes.
Then when somebody tells me endophyte, my thinking is that something is inside the plant.
Yes, and the soil as well.
But you are saying it's in the soil.
Those will be the...
Look at that picture again, because what you are telling us is different from the definition of an endophyte. Okay, thank you.
Another question?
Good afternoon. I just have a question based on what the previous speaker was asking, is that did you inoculate the endophytes at seed sowing, or did you do seed priming, just for clarity?
Did I seek?
What I'm saying is that for confusion purposes, as the previous speaker was saying, I wanted to know, because you are saying that your endophytes, they do promote growth, right?
Yes.
Yes. So I wanted to know that when you do your endophyte application, based on the pictures that you showed us there, was it done at planting, or was it done through seed priming?
I was only reporting now on a review we're working on. It's not a study that was done by our group.
Oh, okay.
But in any case, I want to believe they were checking for growth promoting factors there from the plant and the surrounding.
Thank you.
Thank you, Dr. Mameja. Okay, colleagues, our next speaker, it will be Ingaba. Anyone from Ingaba?
Is a claim in
Yeah.
which laptop this? No,
Okay, good afternoon, everyone. As you can see there, my name is Acclaim Mwila, and then I'm the application specialist for Ingaba Biotech, responsible for our genomic services, as well as the Bio portfolio. So what I'm going to just go through with you today is just highlight some of the relationship or partnership that we have with Unisa as well as Africa BP, and also update you on the latest technological advancements that we have put in places for genomic analysis. So as you can see there, that's pretty much our headquarters in Macleneck. So we actually expanded the whole place. I think we soon going to be call it in our campus, because the whole of the area there, we're just opening offices and offices. And then just again, we've recently opened official offices in Kenya, in the wetlands, as well as in, I think the Nigeria one is quite old, in Ibadan, opposite to IITA, for those that know where the IITA is. And then one slide that or picture that I haven't put is also the office in Tanzania, in Dar es Salaam. And then we are working on opening new offices in Senegal, Cameroon, and other areas in sub-Saharan Africa. Again, just I've mentioned earlier on, we work closely with Africa BP as well as Unisa. And then this is some of the workshop that we were doing in Kenya in December. I believe there is another second edition of the workshop here in South Africa, which will be at Unisa in September. We will also take part in there. And then, as some of you would know, we run what you call Ingaba Biotech Africa BP Genome Challenge, where the participants across the African continent kind of submit their product in a hope to win free sequencing. And I think 2 or 3 weeks ago, we've announced our winners of this year's competition, which is a group from Northwest University at Potchefstroom, and they will be able to sequence, I think it's the horsefly for free on our systems. And this is an ongoing kind of contribution from us in terms of making or contributing to Africa BP's goal of having to sequence many genomes across the African continent. This is the little part that we can play, actually. But it's actually a fun game here as well to play. So just to highlight some of the instrumentation and that are available at our premises across the sub-Saharan Africa for genomic analysis. As you know, most of you get the oligos from us. I think the turnaround is usually 48 hours for standard oligos. And we're able also to produce the probes, but the list for probes is really still very much limited, but we do, for those probes that we can't synthesize, we also have a supplier that is in place for us to get those that we can't produce. And we also do Senga sequencing. I think it was after oligos, we introduced the Senga sequencing at the start of the company. And then we have 2 platforms that do 24 injections, the 2 ABI 3500s, and then we have also the ABI 3730 XL, which has 96 injections. And then, of course, the lab in Nigeria now is fully functioning on the Senga site, and they will soon be able to offer NGS sequencing for customers that come from the West Africa side of things. And then we also have the MySeq, the NextSeq 500 from Illumina, which pretty much soon will be phasing out. And then in terms of the genotyping and the microarray, we have the Agena mass array, which we do for targeted SNP genotyping, as well as targeted methylation analysis. So basically what happens here is if you want to detect SNPs, you know where they are. For instance, you have the RS number or you have a sequence indicating where the SNPs are. We can design an assay for us just to call that particular SNP. Similarly with the methylation analysis, you have a fragment or a gene that you want to do methylation quantification in that specific region. We can design an assay just to do methylation quantification in that particular region. And then we've recently added the microarray, the Thermo Fisher Titan microarray platform for high throughput analysis. So we can do a variety of methylation, genotypings, could be bovine, or even human chips are available. It's a wide variety of offering there. And then in terms of the NGS, we obviously have in 2020, we've bought the first SQL 3E system from PegBio, and obviously now, as you know, with the Unisa, we've also partnered again which I think there will be an announcements in due course. We have the latest, the review system, which we are currently running, and then in addition to that, we have the short reads, the Onzo system from PegBio as well, which is fully installed as well. As you can see there, it's quite a big instrument, the review platform, and those are our 3 musketeers that are really behind the sample prep, making sure that the samples that are sequenced there are of proper quality, the QC and everything. So they're of our technicians there. So the review system, how it's different to the previous SQL 2E system is that we can run 4 cells simultaneously at the same time and get data in about 24 to 30 hours all across 4 genomes. So that allows you to do multi-omics in a single run. Compared to that, to the SQL 3E is that you run the smart cells in a sequential manner. So one smart cell would run, complete, and then the other one will take over. Whereas in this case, you sequence them simultaneously. It doesn't matter whether you have a whole genome sample on one cell, transcriptome, 16S or ITS, and single cell on the other one. They will run all at the same time. So you get your data output across the 4 smart cell at the same time. So that allows us to run about between 8 and 12 smart cells in a week. And then just to touch you again of how the review as well as the SQL 2E work. So we pretty much, you'll hear those that are familiar with PegBio technology talking about the hi-fi reads, hi-fi data. So basically how we produce this hi-fi data is that we take your high molecular weight genomic material. It has to be specifically high molecular weight. So you can use your QIAGEN or Zymo kit to extract DNA that you're going to run on long reads. So it has to be specifically high molecular weight. So majority of your fragments, when you run it on the tape station or fragment analyzer, majority of the fragment have to be 40 kb, 50 kb, or 60 kb long. So that allows you to share to the required size. So basically we take That high molecular weight, we mechanically shear it into roughly 15 to 25 kb long fragments. And after that, we ligate smart bell adapters. It's these loop adapters. And the second step will be to ligate a sequencing primer as well as the polymerase. So this happens on your work desk. It's not something that happens within the instrument. So you anneal the sequencing primer and bind the polymerase on your desk at room temperature. So what you take into the machine, it's a polymerase template complex. Now during sequencing, basically you get this structure now opening into a circular structure, which then polymerase goes around that molecules multiple times, sequencing both reverse, forward, and both adapters over and over and over and over again. And as you can see with polymerase, it's not a perfect molecule, it's not perfect, so it will introduce these random errors, what you see in the red dots there, because that's how the polymerase is. It's not perfect, but what is great here is that if you sequence the same molecule over and over and over again, and because these errors are randomly distributed across the molecule, when you kind of collapsed put together all those sub-reads to create one consensus read, you literally flush out all those random errors. And then what you end up is one high quality, high fidelity sequence, which is 90% of the bases in that are over Q30 plus. And that is where the difference with other technology comes in, with other long read technology comes in, so that we are able to sequence the same molecules over and over and over again and give us high quality molecule. Again, so this is our ONZO system. Our short read platform is also available in our lab, which will allow us to do short reads similar to Illumina in terms of the read lengths that you get, but the technology it's different, completely different. So what you see here, so this is the sequencer itself, and then what you have here is a cluster generator. So basically the clustering happens outside. It's external clustering. So this allows you to cluster a flow cell, put it into the machine, and while the machine is sequencing, you can cluster another flow cell. So it goes on like that. So this is actually the actual screen of the system. So, but what is key here is the percentage of the bases in Q40 plus. So you can see that it says 93.44 of bases are in over Q40 plus. That is really high quality data set there. And then this is the standard that we say you will achieve. 90% of your 4 bases will be in Q40 plus. So that means 100% of your data are already in Q30 plus. And then the flow cells are in, so we have a flow cell that has 2 lanes, 2 independent lanes. That means you can put one sample type on one lane and put another sample type on the other lane, and then run them independently. So this is a very key differentiator of our ONZO system here in terms of the quality. It's the highest in the market. How it's happened, so we use the sequencing by binding chemistry. How is it different to your standard sequencing by synthesis chemistry? So basically what happens is we separate the interrogation and the activation steps. So with your sequencing by synthesis, basically you have irreversible terminator molecules that are introduced into the growing chain. And when the light is emitted, we capture that light and we call the base, and then we just cut off the fluorophore to activate the binding site on the growing side, side of the chain. And that's how it happens. With the SBB, obviously, we have the molecule that is blocked. We introduced a fluorescently labeled nucleotide analog. And then the polymerase will capture that an analog. The light will be emitted. And once that light is emitted, we know what base it is. We don't keep that, the system doesn't keep that fluorescently labeled nucleotide analog. It actually releases it because it already knows what a light has been emitted, and that particular light corresponds to a certain base. And what comes in then now, after this molecule has been removed, the site is activated, and a native nucleotide corresponding to the light that was emitted is incorporated into the growing chain. So we are literally binding a native nucleotide, and not the one that it's an analog with the fluorophore molecule on top. And that's where the the quality difference comes in. So with the system, you have two flow cells. You have 200 cycle flow cells. You can do 1 by 200 or 2 by 100, and then you have your standard 300 cycle where you can do 2 by 150. And then, as I've mentioned, 90%, over 90% of your bases are in Q40, and then other libraries that you prepared for Illumina or Ion Torrent can be sequenced on the same platform. And then that is our Gene Titan for microarray, where we do all the microarray stuff. And that is our the mass array system, the multi-TOF-based system. Yeah, thank you.
Thank you. The technology changes every time. You must keep up. Any questions for Atling? Going once.
At the back there.
Yeah, thank you. Maybe out of curiosity, I wanted to find out if you have ever had, let's say, difficulties when you were sequencing maybe marine species. Have you ever maybe done sequencing on the marine species? Because I once sequenced the abalone, and it was so difficult for me to get, let's say, good quality rates out of PacBio. So I ended up maybe settling for 5K instead of 20 KB of the data. So I just want to find out from you guys, maybe if you have once maybe had a challenge when doing maybe sequencing on the marine species.
No, we haven't done any marine genome except marine microbiome. So we're able to sequence and produce data for marine microbiome, but not individual species. We haven't done that yet. Yeah.
Okay, another question? Okay, thank you, Atling, for always updating us.
Yes,
on
you're welcome.
The technologies you have. Thanks.
Good afternoon, everyone. I want everyone just to stand. And we're going to follow Prof. Casper's advice yesterday, because he said dancing is so important. Let's just stretch a bit before we go on. Just get our legs and everything loose a bit. Okay. Our next speaker, I'm sure I have no introduction needed. It's Professor Pillay. Now, Professor Michael Pillay is a distinguished professor at the university with more than 35 years of experience. In the education sector, he has supervised over 30 master's and doctoral students and has taught several courses in biotechnology. In addition to his teaching and supervisory roles, Prof. Pillay is a professional language editor and an expert in science writing. He has published over 100 referee journal articles. And if you do the maths, that is quite a lot. Okay, well, Professor Pillay, please take a stand, and if you would like to listen.
Okay, thank you.
Thank you for the introduction. I got my PhD when I was 41, so those accomplishments were done in the last 33 years or so. Thank you all for staying late. This is almost like the graveyard shift, so let's see what we can do in this. How many minutes do I have? 20 minutes or so. I want to talk, I think the last time on the 22nd of May, I spoke about banana breeding. And the last speaker from Inkaba, I can't say that word very nicely, he mentioned IITA, which is the International Institute of Tropical Agriculture. The head office is in Ibadan, Nigeria. I was based in a substation in Port Harcourt, in Oni, and that's where I spent 3 years and learned some of my banana breeding. And then I was moved over to Uganda, where I spent 6 years also doing banana breeding, cytogenetics, and molecular work. Can bananas play a role in food security and poverty alleviation? Those two phrases, poverty alleviation and food security, are almost like hackneyed phrases. They are almost like cliches. Every country you go to in Africa, they always talk about food security and poverty alleviation. Although they are regarded as such, they are still relevant. I think I heard over Radio 702 yesterday or so, they said 20,000 children in South Africa go hungry to bed every night. So they are still relevant terms. So I have a project which I wrote for a niche area project, but I haven't received any funding so far. I keep hearing about certain amount of funding ring-fenced for the niche area project, and I had Prof talk this morning, he got 5 million or so. If I get 1 million, I'll be too happy out of that 5 million. So, on Monday, it was 6 degrees centigrade, and you can see one of my cats taking some heat from one of my oldest dogs that I have. He's about 14 years old now. And when I look at this picture, we always say they're fighting like cats and dogs, and I think that kind of phrase loses its meaning when we see this kind of pictures. So, if I go back to my topic, I think the topic is reasonable, and it will be feasible to do if I really see this kind of bonding between animals. I taught biology and general science for 10 years. They always say, start from the known and go to the unknown, and that's what I want to do. I want to start from the known. This, by the way, is a banana plant. Banana is not a tree. If anything you learn today and leave this place, please learn that banana is not a tree. It has got no woody tissue. Banana is a herbaceous plant. Okay, it's a herb. It's a monocotyledonous herb. This part is represent what was the female flowers that become the fruit. It's a parthenocarpic plant. It does not need pollination to produce the fruit. This part that hangs here is the male part of the flower. So even in bananas, you can see the male part always hangs. These little babies from bananas, they are known as suckers. And sometimes you hear people say, bananas suck. Maybe it's true because they produce suckers, but that's not what I'm really trying to tell you. I think in South Africa, when I grew up in South Africa, I only knew one variety of banana. What you see here, this is a Cavendish variety. It's only when I went to Nigeria and started working on bananas, I realized that there are over 1,000 different banana varieties in the world. So all of us are familiar with this. This is the Cavendish variety. Okay. There's the banana plant again with its babies, the suckers. This is the male bud. From the male, I want to show you the different parts of the banana plant that is useful, that is edible. And that's the crux of this project. The project is to show that green bananas can be eaten. So we're not going to talk about ripe bananas anymore. Green bananas and different parts of the banana plant can provide food. And that's when I talk about food security. I did a similar project in Malawi, Tanzania, and Mozambique. In Malawi, for example, they depend on maize. Okay, you blame the US government for that. For introducing maize and pushing maize in all of Africa. But when there's no rain and the maize crops fails, then people say we are starving, we have no food. But if you have about 5 or 10 banana plants in your yard, banana is non-seasonal. You don't have to plant every year. It will grow, it will produce suckers, multiply on its own. Very little labor is required. You only need labor in the very first instance when you're planting it. So when I read about agriculture in South Africa, especially the small scale or subsistence farmer or semi-subsistence farming, most of it is done by the older people, the female and the children. So banana can save labor costs. Once you plant it, you don't have to plant again. It will keep growing. There are several different parts of the banana plant that can also be eaten. The male bud, yeah. There's the male bud. You can get the male flowers, and you can make a good stir fry with the male flowers. The central part of the banana stem, it's called a pseudostem. It's not a real stem. The central part, if you get the squash cylinders there. If you go to markets in India, you will find this. And this is a dish that you can make from there. Usually in South Africa, once the bunch is harvested, this is chopped off and everything is thrown away. But there's food. That's why I want to introduce this project. There's several different forms of food that you can get from the banana. This is a dish here from Tanzania, banana and meat. Indizi nyama. So even there, nyama is meat even in Tanzania. Okay? Here, banana is also mixed with meat. This is a common dish in Malawi. I think Prof. Ngambi is not here. He's from Malawi, I believe. You can make banana patties from green banana. And if you are vegan, there's something you can to live on. And if McDonald's wants to make vegan burgers, you can use banana. There's a patty. You can just put that into a burger bun and get a burger. So there are several ways in which you can use bananas. Another type of banana is a plantain. They are more common in West Africa and very common in Nigeria, plantain. It's a much larger banana. The only difference here is that when the plantain ripens, it still remains starchy. You can eat it as a ripe banana, but you will actually feel the starch in your mouth, okay? And this is a delicacy in Nigeria, and even in West Africa, okay? Fried plantain, fried in palm oil. And here's some jollof rice with the fried plantain. If you go to Uganda, I used to pass this market every morning on my way to the banana research station, and you will see truckloads of bananas coming into the market. And when I used to drive past in the afternoon, you'll hardly see anything left, okay? And banana is a staple food in Uganda and several other countries in East Africa. In much the same way we take rice and put in our plate, or we take pap and put in our plate, there they use banana. The green banana is steamed and then it's mashed, and it's called matoki, green banana. It's eaten with any kind of stew. You know, Lake Victoria is nearby there. You get a lot of tilapia. Those are huge tilapia. Not the tiny ones we find in South Africa, these little ones. There, you get very big tilapia there. 10 minutes left. Thank you. I think I will. There's some banana with beans. This is the matoki that I'm talking about that they eat in Uganda. On the roadside, people fry the bananas or barbecue them. Next to this, you'll find a person barbecuing some goat meat on a skewer. So for lunch, you can stop by and have some roasted banana and your roasted goat meat. And if you are thirsty, you can even get banana juice. And you know that if once you produce juice from any plant, you can ferment the juice and make anything else you want, any other kind of drink you want. Okay, there's banana gin called waraji in Uganda. No, too fast. These are just an example of the several products that you can make from banana. I think common banana bread you can always buy in South Africa, but banana chips you can get in Woolworths and several other stores. It's a big thing in India, banana chips. If you're walking along the street, you will see this guy with a big container frying his banana chips. And you can buy fresh banana chips and for 20 rupees or 10 rupees and eat along the road. Okay, so there's several ways in which you can use bananas. There's, what's this? Cake, banana jam, banana juice, banana wine. There's banana beer. There's a person in Tanzania, in Arusha. She makes banana beer. It's far cheaper than buying Carling Black Label or anything else. So if you want to get high, why spend money on spend 2 rand or 3 rand on Carling Black Label? You can spend 50 cents and buy banana beer. One very important byproduct of banana, which is not used in South Africa so far, is the banana fiber. Once you can get banana fiber, the whole new world opens up to you. You can make several different kind of products with the banana fiber. There's a machine that you can use, and they are available in India. I think for about 50,000 rand, you can buy a complete machine to get the banana fibers and make other things. So this was included in this project as well. And once you have banana fibers, okay. These are some of the things that you can make from bananas. If you go to most of the tourist sites in South Africa, you will notice that people are normally selling wooden stuff, wooden elephants, wooden giraffes. Those things are not even made in South Africa. Someone told me they come from Malawi. But there's an opportunity to make things directly from bananas and sell them. Fill those stores. And is there a time lag with this one? No? Okay. There's a picture I took in India. You can even make very sophisticated handbags from banana fibers. So once you have fibers, you can make several things. They even make saris in India with banana fibers. I even bought a shirt, banana fiber shirt. It's too cold to use it today. It's a summer wear. All right, so green banana flour is a product that is very, very useful. High fiber content, resistant starch, vitamins and minerals, but I just want to hone in on the most important thing here. It has a very low glycemic index. So banana flour is good for people with diabetes. Long time ago, I think I read, they said that by 2015, 1 in 3 people in South Africa will have diabetes. I don't know what's the new situation now. What's Jasper, do you want, somewhere, maybe 1 in 5, 1 in 4? So banana flour is good for diabetics, and you can make a lot of things. You can make banana roti, banana chapati, all of these things, flatbread, once you have the flour. I just saw this yesterday on Google. Stop throwing banana peels, and you can see all the different things that you can use banana peels for. So it's a source of fertilizer, teeth whitening, shoe shine. Apparently soap, you can even use banana peel as soap because of the high potassium. I told you that we only have one variety of banana in South Africa at the moment. This is highly susceptible to a disease called Fusarium wilt. And the disease is right here in Mozambique at the moment. Someone has to carry a diseased plant into South Africa, and that will be the end of our Cavendish banana. So this project, I told you earlier on, there are thousands of different varieties of banana. There's some examples here. There's also a banana that looks like this. They call it the praying hands banana. Can you see why it's called praying hands? Okay. But basically my project is to bring in new varieties of banana into South Africa to replace, not to replace this one. I'm not sure if the commercial farmers will be very happy with that story. Create a demonstration plot somewhere in Nelspruit, because there's a partner there from ARC Nelspruit. Select farmers from the surrounding villages, train them on banana production, and then give them the planting material to establish their farms, and then give them further training on banana production and all the post-harvest products that you can produce from banana. So that's basically this project about. I'm trying to write a grant for some, I think it's due end of this month, for the first phase, which is establishing the demonstration plot. Do I have some more time? Let me see what I have next. So some advantages of banana cultivation, I already said that, you know, it's a year-round production, you plant it once, allows intercropping, because bananas are planted 2 meters by 3 meters. So the space in between. You have to go to Malawi to see how they use space. In between the banana, you have your pumpkin, your maize, your beans, your sweet potato, everything planted there to save space. And this is the benefits of the project. I want to give them to small-scale farmers to create niche markets. A lot of sustainable development goals can be fulfilled with this project. Hazy View is the location. There are several partners from Unisa who are interested in this project. And as I said, I did this project before. There's a paper that came out from Mozambique. And what else do I have here? Yes, this is what I would like to see one day. And when we reach this stage, then all of us will go there and sing a song. Someone told me time is up. Nobody's asking me what song. Work all night and drink a rum. Cut banana till the morning comes. Come, Mr. Charlie Man, Charlie me banana. Daylight come and me wanna go home. You have to be a certain age to remember the song sang by Harry Belafonte. Thank you very much. If there are any questions, I will try to answer them.
You will allow 2 questions.
There's Sibusiso.
Thank you, Prof, for a wonderful presentation. I'm just curious, from these species, different species of bananas that you've spoken about, which ones are the tastiest?
The tastiest banana is a variety called Sukali Ndizi. It's a smaller banana, much sweeter than the local variety. If you introduce that banana, everyone will buy it. It's a smaller banana. Sometimes you see this very large bananas. You wonder, should I eat this in the afternoon, whether it will digest. But Sukali Ndizi is a very sweet banana. The same one, it's called a different name in India. It's called Rastali in India. Yes, sir?
Okay, thank you, Prof, for the insightful presentation. Okay, so I picked in your presentation that most of the value addition is focusing on green bananas.
Yes.
Is there a reason for that, why we cannot implement the same processes on ripe bananas? And the other question, I just wanted to find out why at the moment we only have one variety in South Africa, or most probably Southern Africa. Is there a reason, or possibly we might want to introduce these varieties when there are issues, or they may not grow in the climatic conditions that we have here?
Okay. It's a common sweet banana that's even grown in South America. You see, before the Cavendish, there was a variety called Grand Nain. And Grand Nain was overtaken by a Fusarium wilt, tropical race 3. And that's when they introduced Cavendish banana. So because it's a common banana that is grown in South America for export, South Africa even adopted that. So I'm not sure why no one thought of bringing the other bananas to the country. But what I'm going to bring is the cooking bananas and the sweet bananas and the plantain. So why I was emphasizing the green banana is that in South Africa, I think if you tell people that you can eat green banana, they look at you 2 times. Everyone only knows about I grew up in a banana farm. I didn't know that one day I'm going to work with bananas. But when there was little to cook, my mother took a walk in the banana field, came back with green bananas, and prepared a dish for us. So it's an edible thing. I don't know why in South Africa no one wants to eat green banana. So I want to emphasize that part. But I will also bring the Sukali Ndizi. You can eat it as a ripe banana. And the East African island banana from Uganda.
Prof,
Yes, sir.
just to follow up on the issue of one cultivar, one variety. Is it the commercial one? Because I know, I'm not sure if the guys from Venda and Dr. Mahali, we have several bananas that I've seen at home. So I'm not sure if it's just the one you're talking about is the commercially available one, or the other ones you're not sure.
I think we have 1 or 2 other varieties. I was making arrangements with one of my students from Toyandu to get me some of those other varieties and try to, because I think Prof. Titus is interested in the what, you know, bananas can stain your clothes. You get a dye from that. I think he's interested in that. So I've got one wild variety growing at home. I want to bring that. And whenever I go to Toyando, I'll bring a few others. Okay, thank you. Thank you very much.
Prof, I had a colleague at UKZN. She froze her bananas and made lollipops out of it. Yeah, so there's another one.
I think I only showed you part of the things that you
Yeah.
can make from banana. Yes.
So, thank you for a very interesting talk. Our next speaker is someone you also should know and must know, is Dr. Leslie Adriaanse. She's a college librarian at our university and holds a Doctor of Philosophy degree. She has authored 20 publications. She has garnered 4,377 reads and 340 citations. Her research interests include promoting e-visibility for researchers, research metrics, and the relationship between alt metrics and bibliometrics. Welcome, Dr. Adriaanse, and the floor is yours.
Thank you. Good afternoon, everybody. I have the graduate session. All right. Let's just see. Okay. Sorry, I have to get this going now. Okay. All right. I take cognizance that I'm the last, and I'm the one between you and the door. So I'm not lunch. So what I'm going to do is I'm going to skip through. I understand that the presentation will be shared with the attendees. So there are sections that I'm just going to rush through. If you have any questions, you can ask me afterwards. There's a section that I would like to spend a little bit more time on, so please indulge me. All right. Right, so we're just going to quickly go through. We've got general information, accessing the Unisa collections. We're going to look at, oh dear, sorry. The importance of research impact and research e-visibility. And we're going to look at the research impact and performance, strategies to enhancing impact of your research. We're going to touch on predatory publishing. I get a sense sitting here today that there's a lot of questions around the quality, high-impact journals, and of course also predatory. MDPI was mentioned. I'm not going to go there specifically, but I'm going to touch on some of the red flags. Quality open access journal selection. I want to spend quite some time on that, if I may. And then, of course, transformative agreements. And then we're going to touch on research e-visibility. All right. I'm not going to spend much time. It is going to be sent through to you. I just want to make mention that we've got the 2 emails up there. Please take note. We have a Unisa library authentication system in place. I know it's not the best. I take cognizance of that. But if you are confronted with these 2 screens, you need to use either your Unisa network login and password, or if you are a student, you need to use your My Unisa login and password. If you are not getting access, if you're using your password past these, please use the emails that I've given you there. We need to know that you do not have access. I think it's the most frustrating thing if you have access but you haven't got access. So please let us know so that we can help you through that process. All right. I'm just going to quickly go through the library webpage just to show you the various services as a postgraduate student. I've given you the email address. You can link through it from Unisa online. There's various services. As you can see, the very first top half is our discovery tool called Summon, and it's a wonderful tool. It's a new tool, so if you're having any issues with it, please let me know. But what it basically does is it's software that runs across all of our databases that we are registered as an institution. So it should give you access. If it's not, please let us know. Again, it's something that we need to know. Then we've structured the webpage in such a way that we are highlighting services per client group, but then also you'll notice for any of the clients that have special needs, please. And we've got quite a few of our clients that come back to say, you know what, we weren't aware, but if you do have a special need or you are aware of a student or a client, please let us know that we can assist our clients. All right, and then I'm just going to move on. All right, there I've just given you a screen print as a postgraduate student of all of the services that are available to you. I am basically the liaison between the college and the library. So if you are not sure of any of the services, please contact me. And there's another thing that I just need to mention, that as I become aware of students that are registered in the college, I receive a list and I upload your email onto a MS Teams channel specifically for postgraduate students. So if you are not receiving any notifications from me, you need to let me know, because that's where I'm going to be advertising, promoting events, training sessions, etc. So please, if you are not on my distribution list, please let me know. And also with staff, I'm not always aware of all of the new people that come in and leave, so just please let me know. It's an open invitation. All right, we're going to move on. I'm not going to go through this. It's basically just screen prints taking you through how you can search for items on Summon. So if you have any queries, please, time is running out, and I don't want to keep you here too long. Basically what it boils down to is you can do the search. So this is just to show you that you need to authenticate as soon as you get onto a link. But we have the options of access to PDF or the full text. Again, if you are not accessing the full text, you need to let me know. All right, so I'm just going to, this is just to show you how you can access and how the information, how you can, once you've got the information, you can either email it to yourself or you can export it to an online reference management tool. All right, so I'm going to move on. All right. You need to know about the institutional repository, okay? It's, forgive me for saying it, but it's the Unisa Library brag bag. So what we are doing there is we're trying to upload any of the research that is initiated and published within Unisa. So it's anybody that is publishing, you can then upload it onto the institutional repository, given the copyright restrictions, things like that. If you're not sure, contact me. But it is also the let's say platform that we use to upload the completed electronic or the masters and theses dissertation. So please just take note that for any of the postgraduate students, what we've uploaded is the procedures for submission, and of course if you've got any supervisors that are external and they're not sure, we've given you guidelines there, procedures. Okay, is it not working? Oh dear, why is it not working? Oh dear. Okay, there you go. There we go. All right, just to show you, we've also got the Unisa M&E policies and procedures and guidelines. And that's just to show you the platform to the institutional repository with all of our collections. And then, of course, you'll see the electronic theses and dissertations. Now, many of the students would contact me to say, you know what, I need an existing example of one that has been handed in. That's where you can go and have a look for it. All right. I'm not going to spend too much time there. Why is it doing this? Sorry, I don't know why it's doing it. Where am I? Current slide. Apologies. It seems as if I've clicked on something that I shouldn't. Okay, there you go. All right. You also need to take note of the Unisa Research Data Repository. Please, there is various, I would say, information and guidelines that you need to take note of. I've given you the address. You'll also see here the Unisa Data Management Policy. Please take note of that. If you're not sure, you're also welcome to contact me so that we can take you through the process. And then, of course, you've also got our actual repository where we have our data and we've got items. So, and you'll see we also partner with Fixshare with the data repository. All right. Accessing the Unisa library services via the LibGuides. It's an alphabetical list. A lot of, we've got generic LibGuides, we've got subject LibGuides. You simply scroll down. It's an example of one of the subject library guides. Okay. I need to mention, I get so many queries from people saying that they make use of Google Scholar. They're very frustrated because they are confronted with having to pay. You don't need to pay. What you can do, I've given you screen prints here. You need to go into Google Scholar settings and you need to set up the library links. And what it does is once you've saved the setting, it allows you to access, if you're searching for items on Google Scholar, you'll see if it's within our library collection, it'll show you that the Unisa library has subscription. And of course, from this point onward, you also need to authenticate. Okay, so I'm not going to spend too much time. I just wanted to give you, because that's one of the biggest queries I receive. Okay. Quality journals. I'm going to try and take you through. I want to give you context why your research quality journals play quite a large role in your performance and the research performance impact of the institution. So it is generally accepted that the research performance of researchers are gauged, evaluated based on their research output. We've heard quite a few distinguished scholars here with quite a list. And the citation counts at these, and especially on the citation resources, it receives quite a lot of attention. So, research indicates that publishing in a quality journal. Now, I didn't list all of the research that indicates that, but publishing in a quality journal increases your citation counts. Quality begets quality. In other words, somebody that's really wanting to make use of your research, if you've published in a quality journal, they're going to be using it because they want quality research. So it's basically building on the body of knowledge, using quality research for your research. Okay. The reputation of a journal also plays a major role in the researchers searching and utilizing research, citing research in journal with a good reputation. Oh, sorry. I think, yeah, it's, okay. So, Unisa has now got what they call a high impact journal framework that they've put in place. In other words, where we're trying to establish best practice, good practice, publishing in high impact, good quality journals. And of course, this is going to allow high citation counts per journal, per article. And of course, it depends on, of course, the discipline, but we've heard previously quite a few high-impact journals like Nature, etc. Research indicates citations are significantly increased in open access publications compared to the publications behind subscription paywalls. Why? Because it's available. It's I've got 5 minutes. All right, motivation. You want to be publishing in a quality open access journal because you're preventing predatory publishing. Research sharing and dissemination to a wider research audience. It's given. Articles in open access are available immediately. In other words, as soon as they are published, within 24 hours, they are available for a wider audience to cite, to make use of. All right. And of course, are increasing your e-visibility as a researcher. You also want accurate, these citation resources allow for accurate and relevant research metrics. Quality control within academia, peer review. So if you're publishing in a quality journal, you know that there's peer review in place. And of course, you want digital preservation and consistent, persistent archiving practices, which is, that goes hand in hand with quality journals. And of course, right, if nobody knows that you have an article, how can they cite you? Okay, so we're going to touch on the impact, so please indulge me now. Researchers need to promote their research in order to increase their citations and increase the impact of their research. What does it mean? What is the impact of your research? Okay, so what I'm going to do now is I'm going to put it into context. I'm using that Times Higher Education rankings, and we all know rankings, universities. This is basically what they're using. So 59% of the Times Higher Education university rankings is based on the research environment and research quality. And you can see the citation counts of the faculty and the academics of the institution makes up to 15% of the ranking of a university. Okay, so I've just made a screen print. I'm not going to spend too much time. You can go through it later on. But it basically boils down to, this is the formula that they use. And as you can see here, the 59% research environment and research quality. But it all boils down to citation impact, seen as important, and it speaks to the citation counts and the research influence. In other words, the average number of times the publications are cited by other researchers, and it helps to show how the university is contributing to the subject discipline, but also the body of knowledge. Okay, so this is just a screen print that I've used the Elsevier Syval benchmarking tool just to give you an idea. At this point in time, this is the latest now, Unisa currently has an average of 7.6 citations per publication. This is across all of the disciplines. So basically, what does it mean? It means that last year we had 7.2, so there's an increase. That's exactly what we want. We want more citations per publications. Why? We're looking at rankings ultimately, okay? So at this point in time, you're looking at a field-weighted citation impact. It's an indicator. So it basically boils down to anything above 1 means it's above the world average, which is good. That's exactly what we want. So we're wanting to look at strategies. Now, you can see varying through COVID. I've been keeping track of it to show there has been growth. Sometimes it takes a little bit of a dip, but this year it looks as if it looks very promising going into from January to June. Okay, so we're looking at strategies for the publishing in quality journals. Why? You're looking to publish in high impact, open access. You want to prevent predatory, and there's all sorts of strategies there. Ultimately, these are the 3 that I can, as a librarian, can assist you and support you as a researcher in enhancing your research performance. Okay, definition. Generally speaking, predatory journals, it's publishers that exist for the sole reason to profit and not to disseminate high-quality research. Unethical pay-to-publish practices by publishers and editors masquerading as serious, legitimate, scholarly entities. I could not resist that picture, because that's exactly what's going to happen if you do not understand the perils and the consequences of publishing in predatory journals. Okay, so let's just... Why are and I've given various reasons here, from researchers, especially the developing countries, especially the emerging researchers for prey. It's easily accessible, you know, and it's online environment. Open access, the whole open access environment allows for predatory because it's online. You're not always sure. They can hijack and they can masquerade. Of course, earlier on, there was a mention made about how long it takes to publish. There's a lot of dissatisfaction with the traditional academic journals. Publishing, it just takes too long. It's too difficult. And of course, you've got aspirations. The young academics get anxious. They want to publish as soon as possible. All right. Oh dear, sorry, it's because I am... All right, there's consequences, all right? You do not want to go there, it doesn't look good on your CV, there's no digital preservation, your intellectual property rights transfer from the author to the publisher, you don't want that. Okay, so what I've done is, there's training, there's a whole session that I do on predatory, and we also touch on the predatory conferences where I give you advice how to use and the red flags. I'm including the accepted Ottawa definition, which gives you 4 basic red flags of predatory and suspect questionable journals. Okay, so I just want to unpack, please forgive me. It's the first one is solicitation. I was hearing all sorts of stories of receiving emails, etc. Now, even I fell prey to this, and you'll see here it's in electronics. I'm not an expert in electronics, but I was asked to be a guest editor. It just doesn't make sense. Okay, so these are the type of emails. I put it in as an example. I couldn't resist it. I thought, self-citation practices, it's a huge, huge issue at the moment. You're looking at peer review, where you'll see sheer numbers of publications obviously make it impossible to guarantee minimum peer review standards, things like that. Pool of reviewers, you've heard of all of these things. These are some of the red flags. Then APCs. Now, somebody mentioned a specific publisher here, huge amounts that they ask for as APCs. Go and have a look at the increase. There's some of them that have up to 220% increase in APCs from one year to the next. And yeah, it becomes a huge problem. I've given you here. Article processes, it's levied by journals that have both high impact factors and site scores, okay? What is the driving force? Academics willing to pay high APCs due to misconception of high APCs is a quality journal. Now this is a screen print that somebody made a statement. I couldn't resist it because there's a misconception that if it's a high impact, high site score, it's not predatory. Okay. Earlier on, we had people mentioning the DHET quality, the list. I'm not going to go through too much detail, but if it's listed on the DHET, it's subsidy bearing. Many of them are also on what we call the quality lists. In other words, the Directory of Open Access Journals, you've got Elsevier, you've got the Clarivate suite. They also have suspect. Earlier on, Prof. Rees was mentioning small percentage on Scopus, but it's there. They could also be marginal suspect. But a delisting from these quality does not necessarily mean that it's predatory. It means that it's under investigation. And that is something that the people need to realize. There's a time lag. So when people start saying that there's problems, there's violations, etc., it has to go through a process of evaluation with these products. Now, keep in mind, these are the products that I need to use to assist and support, and these are the products that officially are available to verify status. It's problematic. I'm sorry, please forgive me, I need to go. Delisting is not necessarily, it will still be on the DHET list. Only once it's delisted from the DHET list is it no longer subsidy bearing, but that's the problem because there's a lag. The DHET list comes out once a year. We have now, as a part of the research committee, we have requested the DHET to have more frequent, at least quarterly, evaluations of that list and to subsequently send it out to all of the institutions, because it's a problem, because you've got a lag. It could be that in the time that list was published and officially sent out to all of the institutions, a journal could have become from suspect to predatory. you're not going to know because it's on the DHET list. And that's, many of the people say, but it was on the list, that's why I've used it. And there's a misconception that that is the list to use and it only has quality. Not necessarily. Okay, so typical characteristics, I'm not going to go through all of it, but these are the characteristics given for what we would call a legitimate or a quality journal. Okay. I've added these two screens, this section. These are links to the quality, the criteria used by Scopus and Clarivate to decide on a high-impact quality journal. Okay, so if you are not sure, you can also go and have a look. And if it's delisted from these two lists, it's because they do not adhere to these criteria. Right, okay. Citation metrics, also a problem. I'm just going to scroll through that. Okay. The other problem is that there's no one list that can give you all of the predatory journals. Unfortunately, there is a bit of a problem. So one has to go from one resource to the other resource to the other resource to find. And the other issue that I've also come across, and I need to tell you this, especially with your large open access, and I'm not going to mention names, publishers, if they are flagged as having a suspect journal, they have bully tactics. And they take it up with the various lists, and soon they are removed. Removed. I'm not going to go according to the literature. So if they have an issue with being flagged predatory, they're not happy with that. All right, so quality journals. I'm just going to quickly, the strategy that I would like you to take as a researcher, when you do come across the problem of I need to find a quality journal, how do you go about? There's basically three case scenarios. The first one is you have an existing publication or a manuscript. Okay, there's tools that can assist you, menu-driven, you put in the title, you put in the abstract, and it would then suggest quality journals. Okay. The second one is where you're not sure. Then you need to use quite a few research, or I would say resources. Then, of course, when you have the situation where it has to be open access, it's part of the prerequisite, either as a grant, a bursary, etc., etc. You'll see I've included the journal selection tools here. Then with case 2, you'll see all of the resources that I can suggest as part of your the whole strategy of looking for quality, high-impact journals. The DHET accredited list is here. Cabals. Ladies and gentlemen, we have a subscription to Cabals. It's a very nice tool. I have been trying to promote it quite widely. But what it basically does is it allows you to identify quality journals, but also predatory journals. So just to show you here, quality journal, but what it does with the predatory, we call it predatory reports, what it does is it flags it, and it then lists the violations, which is very nice. So you can actually go and have a look at why they have identified or verified it as a predatory journal. But now the problem is, before it gets to this, there's a gray area where you're not always sure. So what I would then suggest as part of the strategy is this is I would see as a macro evaluation. Then you as a researcher needs to look at what I would call a micro evaluation. Trust your gut feeling. You are the expert in the subject. Go and have a look at previous articles that have been published within that journal. Open them up. Go and have a look at the research methodology. Go and have a look at the content. Go look at the quality of the content. And if you come across any issues in that journal, just stay away from it. It's a dead giveaway on a micro level that something is not kosher with that journal. And it could be that it has an impact factor. It could be that it's got a high site score. You know your subject area. Go and have a look on the micro level and then make a decision. And I usually say to many of my researchers, just your research is so important. It's like you would be purchasing a house. You're investing in a house, you're investing in your career. Everything you publish will be with you for the rest of your research career. That's the same importance and the same priority that you should, when you give to any items that you are selecting, any quality journals. Why? Because it's going to be with you for the rest of your research career. We've heard of quite a few of these distinguished. Everybody, I've published so many publications. How many of them are quality? You want all of them quality. Okay, so, okay. We're going to get there. Various resources. I've included, here you've got journal citation reports. I just want to get to... You've got Scopus. It's a resource that has been mentioned quite a few times. I've given you LibGuides here that can help you, guide you through looking for the quality journals. Open access publishing, exactly what it is, the difference between the various open access directories, and then, very important, please forgive me, but I need to mention this. Unisa has along with SANLIC, we've negotiated transformative agreements. Now, what it does is it allows for some of the publishers have waived all of the APCs. I think Elsevier is one of them that where you can you only pay a percentage. But what it means is it allows for gold open access. And this is a wonderful, wonderful initiative by the publishers and the libraries within the academic libraries within South Africa, is because you are saving huge amounts of money. I've included the LibGuide there, the SANLIC webpage where you can go and have a look. The benefits basically boils down to your research is exposed to a wider audience. The benefits of open access is there for you to see. I've included it here. Okay, we can recap. It's important for you to publish in quality journals. Okay, your research, you've gone through all of the trouble of doing the research. You now need to publish in the best quality, high impact journal that you can locate, that is going to fit the aim, the scope of your research. Okay, I also have training, a longer session, more in depth, that I do give. You're welcome to register. E-visibility, it's very important. Your research needs to be available. Sorry, I'm going. Needs to be available online. It has to be discoverable. You as a researcher needs to be discoverable, even if you are as a developing emerging researcher, just as important for the established researchers. And it basically boils down to the more your research is available online, it can be discovered, it can be downloaded, it can be cited, it can be used. And the vehicles that we use for the e-visibilities, your e-profile. So from time to time, you have been confronted with the ORCID ID. It's part of the traditional profiles. We've got various profiles here, social media, and then of course LinkedIn as well. But basically what it boils down to is specific information. There's no personal information, it's your research information that is contained on these researcher profiles. And it's very important, the ORCID, just to illustrate e-visibility. I've used myself as an example, please forgive. I'm just an emerging researcher. Boils down to, if anybody wants to find you, how they're going to look for you. They do a Google search. So whatever they can find, you need to be discoverable. Your research needs to be linked to your profile, because then your research is accessible. Okay, we've got a researcher here. I'm just going to scroll through it. I'm not going to spend too much time. But it was an initiative that we took with this researcher to increase over time. We try to increase the citation counts and the averages. And as you can see, we took it from 2011, from an average of 2 citations per publication, we took it up to almost 19, which it works. You need to be visible so that your citations increase in count. All right. ORCID. it was put in place to do away with name ambiguity. So you'll see here, this is your basic ORCID, and this is a prerequisite at this point in time. If I understand correctly, you cannot publish. For subsidy purposes, you are going to be required to give an ORCID ID. So please, if you haven't got one, you need to get one. If you're not sure, please get to me. All sorts of information. I'm not going to spend too much time. But just to show you the functionality, again, I've used myself as an example, POPIA Act, but you've got an article, and the journal has now, instead of affiliation, all I had to give was my ORCID ID. But what's nice about it is it becomes what we call a green button on the database. So you see the print format on my left, and then you've got, it becomes a green button. But what does this green button mean? I link. With 2 or 3 clicks, anybody reading that article or accessing me in the database has access to all of my information on ORCID, and then if I've consolidated all of my researcher profiles, what does it do? It links me through to the Scopus author ID or to the researcher ID, which is exactly what you want. You want somebody at a touch of a button to see exactly who you are as a researcher, your interests, but of course your publications, and they want to access it, they want to download it, they want to cite it. Okay, these benefits, I'm not going to go through all of those. The Google Scholar profile, and of course you've also got your ResearchGate. Now these are the basics. So even as an emerging researcher, these are the 3 that I would promote. Simply because it's making you more visible, your research output is accessible, it's downloadable. All right, and all of the information that goes along with it. Recapping. Promoting research output and publications leads to the enhancing research. Your research e-visibility and translates to increased citations. Again, I've done it. You know what? We'll get to this. Okay, all right, right. If you need any additional support, if there's anything that you take away here today, that is my role, that is my primary priority as a librarian and as somebody that understands research. I've been there, I'm doing it myself. I understand your needs. Please contact me if you have any queries, if you have any needs, research needs for support, just accessing the library. Please contact me. Thank you very much for your time and your patience.
Thank you very much, Dr. Auditorium. I know it's information that we need. Sorry if we don't have more time. I will allow one question. It was quite a comprehensive.
I try to squeeze in everything.
There's a question.
So Leslie, you and I have had this conversation a few times, and as you're talking, I'm busy testing things and proving that they don't work. Having worked at UWC and ARC and in Botswana, we appeared to be linked through the university's IP addresses to the publishers directly, and they had their address ranges, and we were trying to get access the other day to Nature Biotechnology, which I don't think we have a subscription to. But my colleague at the ARC literally clicked on that email that I sent him with a DOI, dropped straight into the journal and accessed it directly. And that was my experience of all the publishers. Okay. And we're going to sit in a minute and play, but this is a publisher end process. They register our IP addresses so that they recognize we've got subscriptions, and that doesn't seem to be in place. So can we, quote, can we talk? But
we can talk.
There has to be a way to improve ease of access.
I think at this point in time, we have had servers that have been, there's been upgrades and things like that, and we've been having quite a few issues within this year. So
I don't think it's a Unisa internal issue. I think it's the journals have to, the publishers need to register our IP address ranges.
I think it's an admin functionality that perhaps needs to be activated. But again, most of these queries, as we get as they log the calls, it is something that does go through what we call our e-resources librarian. So I'm sure that they are aware of it. But again, I must be honest with you.
Let's play more. Let's play more.
It's a long day. Back to you on that one. Yeah. Any other? There was another hand here. No hand. Yeah.
Why does ResearchGate say that you have 2,000 citations? If you go to Google Scholar, you say you have 4,000 citations.
Remember, it's 3 different separate citation resources where, if I understand correctly, ResearchGate sources its citations, I think, from Scopus, whereas Google Scholar is a more inclusive, I would say, index to Scopus. Scopus only includes journals and their citations that are included in the Scopus list or the sources list. In other words, Google sources, the crawlers would, it doesn't distinguish between, it hasn't got a specific list. Anything that's on .edu or a scholarly research nature gets included into the Google Scholar Index. So it would include masters and your doctorates. It would include reports, research reports, where Scopus does not necessarily include that. So Scopus is a more exclusive index, unfortunately. So your indexes would differ, and that's of course why the NRF also allows all 3 of the H indexes to be included when you apply, because they take cognizance of the fact that it is separate indexes. So it's not going to, so obviously Google Scholar will have more citations because it's sourcing the citations from masters, doctorates, from research reports, etc., etc. All right, that's it.
Okay, thank you very much. And can we give another hand of applause to all the presenters? And now what we all been waiting for the prize giving. Can I ask Dr. Masebi to come forward?
Afternoon, everyone, or evening. Yeah, I think it's been a long day, a long 2 days actually. They're saying I should stand here. So I think I'm going to try and be very short, very brief actually, because I think it's a bit late already. And I am standing here on behalf of Prof. Mapodi, who could not manage to join us due to other engagements, conflicting engagements. So she asked me to do this on her behalf. So I think what I'm going to do, instead of starting with the prize, I'm just going to do a closing, and then we'll end with the gifts, and then maybe a nice picture, and then we can all go home. So at least no questions for me. I tried to draft something down on behalf of Prof. So just to be brief, I'm really not going to thank each and every speaker by name, but I'll try to summarize. So colleagues, researchers, our distinguished guests, our stakeholders, as we're drawing near, or as we're drawing closer to closing this ceremony, with the theme, I think this was a very interesting theme, growing academia through student voices. I think this is a very interesting theme, and we should actually give a round of applause for all our students who raised their hands really to come and stand behind this beautiful audience and share their research. It takes courage really to do that, because it's not really easy to speak in front of distinguished scholars, because I think often you're thinking, what questions are they going to ask me? So I think let's give them a round of applause. I'm very cognizant of the fact that the room is already empty. We do understand that people had to go to their respective places, but I think they did quite well. Our students, they did very well. So I'm not very good in reading if I write something, but I think this event really was an eye-opener. And also it brings in opportunities really for students, for researchers, to share ideas, and it opens up avenues, for collaborations, which I think is very important. So let us continue supporting our students and also supporting this student symposia, because this is the third student symposia, and I think over the years it was well supported. So let's continue supporting our students and also engaging and sharing innovative ideas. I think throughout the 2 days, you've had excellent talks from our distinguished scholars, distinguished professors, our guests, our external stakeholders who came through to share with us their innovative technologies that are currently available. So I think that is excellent. Let's give them a round of applause, colleagues. And I think also what was interesting, and it breaks my heart because yesterday I could not attend the whole day because of other conflicting activities. But I saw just by the way he was so excited, it really, really showed that the proceedings of yesterday were quite interesting. I think also the students who put up their hands to be part of the panel, I think they shared very excellent ideas. I even heard that Jasper was dancing around, to show that the talks were quite interesting. And I think also for the student to share their ideas, it really helps academics to really see where the gaps are. How can we assist our students? What is it that they require for them to succeed? How can we strengthen our skills in terms of supervision? I'm not really going to dwell into the talks that transpired yesterday because I wasn't really here, but I think in a nutshell, all the guest speakers really shared interesting ideas. I was fortunate today I could sit in, and I think all the talks about the open grant calls for students to, to take part, to grab the opportunities and apply, travel. It's nice being in academia because you get to travel the world. Let's grab those opportunities. And if supervisors are here also, let's share that with our students. I think also the issue that is really troubling for some of our students and researchers as well is the issue of appreciating critics. I think people don't really take it nicely, but I think people should take it as a learning curve to say, how am I going to improve my research? How will I assist my students to also improve? I think there's quite a lot that I can say, but just to summarize and not leave anyone out, our external stakeholders, I'm not sure if they're still here, but we are really, really appreciative of the fact that you always are available to support these events. I think without them, this would not have been a success. So they are always here to share with us also the advancements in technologies to help researchers to see if there's a gap in my work, what kind of tools can I utilize in order to continue with my research. So we are indeed grateful. And then just to re-emphasize our M&D students, congratulations to you all for raising your hands. You are really, really appreciated. And then the program directors. Without you guys, I think some have already left or some were here yesterday, but no longer here today. But without you guys, I mean, you managed to steer this ship. It's really not easy to stay until late, but you managed, and in terms of time as well, although somewhere, somehow, I mean, some of the talks were quite interesting, but I think you really managed to keep with time. And we are really, really appreciative. And then our distinguished scholars, I think it is very interesting to hear your research journeys, you know, the stumbling blocks you've come across through your research journey as well, and also to share insights regarding the research that you're currently doing. I think it helps our students to grow and other developing and emerging researchers. And I think one interesting thing that also came out from today's presentation, I think Prof. McIntyre has already left, the talk that he was giving, it showed how important are the mini projects that researchers are giving to the students, to say it might be a mini project, but all those objectives, you know, they build up to the bigger project. So whatever project you are given as a student, don't take it lightly, because it might contribute, and who knows, one day you might come up with something that is groundbreaking. So that was quite interesting. And then also the audience, even those who have already left. I think you were a very beautiful audience. You were sharing your thoughts based on the presentations that we shared, your ideas, your critiques. I think though that, as I've indicated, really builds, you know, researchers, really also assist people to see where they could, you know, add more activities, how they can build their researchers, and also I think that also opened avenues for collaborative initiatives, because as you're sitting there, you're like, oh, this one is doing this study, and I have a similar objectives. How can I collaborate with those? And lastly, I think the organizers, if you turn on the last page, you will see is it the last page? No, not really the last page. Your hosts are there. Let me take this opportunity to appreciate Professor Ntanganezeni Mapoli in her absence for leading this aspect. I think she has done well over the years. And then also the organizing committee, Lucky, Ray, Neo, everybody. I wouldn't want to mention your names, but guys, if you check on the last page, they are there. I think they've done quite a lot. We've had several meetings, sleepless nights, you know, and sometimes we're thinking they will run away, but here they are, and well done to you guys for hosting this successful event. And to close up, a round of applause, thank you. And thanks to me also for closing this. But before we go, I think to appreciate our students, of course, if we had enough money, I think this is a call for sponsors for the upcoming symposia to say we really need money so that all the students who put their hands up to say we're going to present, we give them something. But unfortunately for today, just only distinguished students, we are going to appreciate them. And let me not also forget the adjudicators. I think this was really tough for them because you need to sit in from the beginning until the end, and you need to make notes, make sure that you take notes for all the speakers and make sure that, I'm not going to talk about Dr. Malubane, but I think they have to critically listen to all the presenters and make notes so that would assist them to say, this presentation was better than that. And then we also appreciate the students who did the posters outside. I think people also had an opportunity to go and check the posters. Now, in closing, I think we're going to close on a high note. We have various students. we'll start with the students. And we have okay. We have awards for PhD students and also for our master's students. We'll start with the master's students. The third runner-up for the oral presentation is Musala Unomukadi. Okay, I don't think she is still here. And then the second runner-up, can we guess? If we still remember the presentations for yesterday. Sibkiso Nhlengetwa. They've all left. And then the winner is Nkhameleng Mukunwana. Shoo, all of them have left. So maybe next time after they present, we'll do the prize giving for day one, and then we proceed to day two, maybe. And then, or if we have enough sponsors, then we do it for 3 days, so that the last day, the third day, it's actually a half day. Then we shall have, Prof, with your banana project, maybe you can assist. So for the PhD students, we have actually 4 winners. We have 2 second runner-ups there. It's a tie. Mr. Makwarela Tsirelezo Goodwill. And Ms. Penny Makumbila. Congratulations. I don't know how they're going to share this. It's not only yesterday in the middle. Thank you. And the second runner-up is Ms. Celia Mozeza. Okay, not here. And the winner is Mr. Busiso Malindita. P53. Congratulations. I just want to see if I haven't forgotten anybody. Okay, so, and just to close off, we also have gifts for our invited speakers. Prof. Pillay, Michael. And then Dr. Leslie Adriansen. Prof. Nkululeko Nyangiwe. If I say Prof, he's going to kill me. Jasper Rees. And in absentia, Prof. Ngambi, I think he gave a talk in the morning. Prof. Bahangwele Masini, I think that was yesterday. Prof. Lucas Neyman this morning. And Prof. McIntyre. And Prof. Claire, I think she gave a talk online yesterday. Yeah, she's actually not in the country. Thank you, our distinguished scholars, and for staying until this late. We really, really appreciate you, and for sharing your thoughts, your journey with our students. We are really, really humbled by your presence. Thank you. And colleagues, I think this brings us to the end of our third symposium for 2024. Yeah. So as I was saying, it brings us to the end, but I mean, I won't be using this microphone if it wasn't for our media people. And, you know, we appreciate them for recording the proceedings, making sure that the slides are nicely presented. For those who had hiccups like Tire with his bag that was just moving on the screen. And then also the horticulture for ensuring that this place is this beautiful. We really appreciate you. And for our marketing team as well. You always show up, you know, to assist us. I always call them Gugu times 2, so they are always available to assist. And everyone who I might have missed, please, I do apologize, but I am saying thank you. And we are officially done. Thank you, ladies and gentlemen.
Thank you.